సర్టిఫికేట్ సర్టిఫికేట్ కీవర్డ్ సర్టిఫికేట్ ఇప్పుడు ఫాలో ఏది వాళ్ళు టీకాలు వేసుకొని లైట్లు వేసుకొని ఫ్యాన్లు వేసుకొని అన్ని ఆఫ్ చేసుకొని పోయినా అవసరం ఉందంటే ఏదో పోతున్నాం సార్ అంటే ఒక మాట చెప్పిపోతే మాత్రం మేము పడుకుంటాము లేదు ఇప్పుడు బీగాల వల్ల ఎంత ఇది తీసుకొచ్చి తెలిసిన ఇప్పుడు పొద్దున ఈ రోజు లాస్ట్ డే చాలా వర్క్ ఉంది నేను అదే 8:30కి వచ్చినాయి ఇప్పుడు ఎవరిదైనా వచ్చేవాళ్ళు నేను అర్థం తీసుకోగలను అర్థం నేను పోతున్నాను అని చెప్పి పోయేటప్పుడు వెళ్ళిపోండి నేను పట్టించుకోను మార్నింగ్ నాకు టైం ఉండాలి కదా ఇప్పుడు ఈవినింగ్ టైం ఎవరైనా నేను కూడా తిరిగి చేసి వచ్చిన 16 ఏళ్ళు బయట తిరిగి వచ్చినండి మన అక్కడ పోతున్నా పట్టించుకున్నాను కూడా కాదు బిగాలే అదే పోతాను అని చెప్పి వేసుకోపోతా ఎట్లుంటుందంటే పరిస్థితి కానీ టీఓటి పోతేనే ఫోన్ పెట్టే అయిపోయాడు ఆమె ఇక్కడ పెట్టేసుకో ఇంకా టీము ఆమె అక్కడ అక్కడ ఇక్కడ ఎక్కడ ఏది ఆమె నువ్వు పోతామ్మా నేను చెప్తా సార్ చెప్తాను కాలంటే ఏదో చెప్తాను టీఓటి నింతకు ఇక్కడే ఉన్నావు ఇంకా నీ డ్యూటీ
ఇప్పుడు పద్దలు ఏడుకు వస్తే ఏమో
ఇంకా నెల ఉంటాయి ఇంకా ఆమె ఇక్కడే ఉండమ్మా ఇంకా ఆమె కింద నువ్వేం పోతుంది ఇప్పుడు వాళ్ళందరూ వస్తాంటే చూస్తూ ఉంటుంది
లేదంటే 10 రోజులు 10 రోజులు చేస్తాను డ్యూటీ 10 రోజులు చేస్తాను 10 రోజులు ఎందుకంటే అయ్యో పద్దెనిమిది వచ్చి బిగా తెచ్చుకొని ఓపెన్ చేసుకొని చేసుకోవాలి ఇక ఆదాయం తెలుసుకుంటున్నాను ఈ నెల అంతా పద్దెలు వచ్చి ఓపెన్ చేసి తెచ్చుకో నాగంతా కావాలి
చూసుకో
పెట్టుకుంటాను
ఆమె పోయిన సాయంత్రం పట్టించుకొని వేసుకోకపోతే ఎందుకంటే ఎవరిదైనా ఆడికన్నా పోని సాయంత్రం పూట మేమే బీగాలు వేసుకుని మేమే పోతాం ఇబ్బంది ఎప్పుడు కొన్నాం రిస్క్ పడినాడు మాత్రం నేను ఇంకా అన్ని బయట వస్తాయి నాకు ఇబ్బంది లేనందరూ సరే హలో 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 గుడ్ మార్నింగ్ సార్ వాయిస్ చూసుకుంటా ఎన్ఆర్సీ కాకినాడ హలో
హలో
ఎన్ఆర్సీ
సార్ ఎన్ఆర్సీ కాకినాడ సార్ వాయిస్ హలో
Good morning, sir. NRCs sir.
NRC తనకు ఆర్వి ఆడిపిస్తే
ఆడపిల్ల మ్యామ్ రీసౌండ్ వస్తుంది, ఒకసారి చెక్ చేసుకోండి. 2 లాగిన్స్ ఉన్నట్టున్నారు కదా, ఒక దాంట్లో ఆడియో పూర్తిగా తగ్గించేయండి మేడం.
ఓకే సార్.
So welcome Srinivas sir. I welcome Professor Srinivas for the online session. So I hope all the NRC, KRPs and the participants have joined and should we have to wait another minute or two. Professor Srinivas, can we wait for another 1 minute?
Yeah, okay, sure, no, definitely. Sure, ma'am. Thank you. You can hear me, right?
Yeah.
Yeah, okay.
Your voice is not actually audible.
Now, is it clear, madam, now?
Meanwhile, you can check your presentation.
Okay, can I share now? Okay.
Yeah, make it bit louder now, your voice.
Okay.
You can check your presentation by sharing.
Can you see my presentation?
Not yet.
Not yet? Okay, just... Madam, I am trying to share now.
Yeah. Have you shared Srinivas sir? Actually, it is not visible to us.
Okay, I will try to... Actually, my system is, I'm trying to add WebEx so that, you know, I can, screen can be shared from my end.
No.
So can any technical person who can help me?
It's not shared.
Sir, first you open your PPT, sir.
Okay, I opened it.
After that, please click on share option. In WebEx meeting, share option is there at bottom side.
Oh, okay. No, so no, yeah, no. See, I opened my account. So over WebEx, so I'm trying to open share.
Yeah,
So it
at,
is
at bottom share option is there. When you
yes,
click,
yes, yes. Yeah, there is a share option. Then a
Uh.
pop-up window came up. Allow WebEx to share your content, and then it is also said grant access in systems preferences. So my open system preferences. I'm just clicking that. I'm using Mac, so I just opened the system preferences. So do I need to change any firewall or anything else for this?
Not necessary, sir.
Okay. So now now I'm opening my. Can you see my screen now?
No, sir.
All right. So that's what. Just,
Sir, please close the pop-up window and again, please click on the share option, share button.
okay. So again, I just clicked. Again, it is showing the same thing. Allow WebEx to share your content.
Yes. Please click on the yes, sir.
No, there is no yes option. It says open
Yes.
system preferences. So it is taking me to the system and then trying to change the system preferences.
Oh.
So I'm using Mac computer.
Sujana madam, shall you continue, madam? Sujana madam, excuse me.
Madam,
Sujana madam, shall you continue, madam?
Sir, no problem.
Okay. Thank you, sir.
Madam, Sujana madam,
Yes, sir.
సార్ దగ్గర నుంచి పీపీటీ మీకు షేర్ చేయమని చెప్పి మీరు ఎవరో ఒకరు ప్రెజెంట్ చేయండి లేకపోతే.
Okay, yeah. Okay. I'm sharing presentation to Dr.
Srinivas sir, your voice is also not audible actually.
Oh, I don't know the
It's
reason.
breaking.
Clear,
Okay,
ma'am.
okay.
Sir audio is very clear. Please check at your side, ma'am.
Maybe issue from our side.
Yeah, because it.
Actually, your
So I'm
voice is
sharing
very
my.
clear, sir, for us.
I'm sharing my slides to Dr. Sujana madam. Yes, I shared my presentation to Dr. Sujana madam through WhatsApp.
Okay. Sir Srinivas, shall I share the screen from my side?
Sorry?
Should I have to share the screen from my side?
Yeah. Yes, please.
Just give me a minute, please. English చేసుకోండి సార్ వాట్సాప్ సంస్కృత్
Sujana madam, please share the SAR PPT in our group. Is there any issue? We will continue.
Yeah, we are doing it.
Okay, ma'am.
I hope it is visible now to all.
Yes, ma'am. It's visible.
Yeah, let's go to the first slide. Srinivas sir, is it comfortable? Shall we go on with the introduction, introducing today's resource person to the participants?
No problem.
Yeah.
So I
Thank
see
you.
my slides here now.
Thank you.
So if you want, you can make it as a full screen.
Just a few words.
So I will say next slide so that you can keep moving slides.
Srinivas, I can't. So
I
thank you
hope-
for giving me this opportunity to introduce the most evident personality of Central University. Professor Srinivas. He is the professor of Department of Animal Biology, School of Sciences, University of Hyderabad. So he has completed his post-graduation from the SK University, Anantapur, in the field of biochemistry. Then he had done his PhD from Banaras Hindu University, Varanasi. So he was a postdoctoral guest scientist in the Department of Development of Evolution in Uppsala University from 2003 to 2005. He has also done a postdoctoral research scientist at Birmingham University, Cambridge, UK from 2005 to 2008. So he is also worked as a research associate in the Institute of Cancer Sciences, the University of Glasgow, at United Kingdom from 2008 to 2011. So later on, he joined as a reader in the Department of Animal Sciences, University of Hyderabad in the year 2011, and he continued up to 13. Later on, he has become associate professor in the same Department of Life Sciences till 2018. Then he has become the head and dean professor of animal biology of Life Sciences, University of Hyderabad. So at present, he has 3 ongoing projects worth of 1.5 crore. He has published more than 20 papers in high impact factor research journals. There are 4 research fellows and 1 DST inspired fellow are working under him, and 1 PhD fellow is working under him. His area of interest is his field of interest is epigenetic regulation of gene expression in mammals during the cellular development and differentiation and diseases. So, thank you very much, Professor Srinivas, for giving us this wonderful opportunity to get to know about your knowledge. And I welcome you onto the session, please.
Okay. Thank you very much, Dr. Sujana, for the nice introduction. I hope that I'm audible to everyone. I see that from Sujana point of area, even her voice is not clear to me. I hope that yeah. So please, I will request as and when I require to change the slide so that you can change the slide.
You just say next.
Yeah. So yeah, sure. Okay. Thank you, everyone. Thank you for the opportunity. Today I'll be talking about eukaryotic genome organization, structure, and function. As all of you know, if you really wanted to understand ourselves, how that life has started, how that life functions are executed in us, it is very important that we need to understand about ourselves. That means understand about how that we were built, and then how that within our building block there are cells, how that genome is organized, how that different genes based on the environmental cues, they are specifically transcribed in a cell type specific manner. So these all basic information is very much important for us being a teachers, so that we can understand about the cellular processes, so that then we can understand the development process as well as the disease processes. So next slide, please. Yeah. See, life of every organism, starts with a single cell, that is zygote, which is basically formed by the union of haploid oocyte, and then haploid spore. And then from this single cell, as you can see that father contributes haploid genome, mother also contributes haploid genome. Thus, it becomes a diploid genome. And then this we call it as a zygote. So, zygote by means of a series of cell proliferation and then differentiation, giving rise to multiple cell types. And then eventually we can see them as a morula form and then blastula form. As you can see within this particular blastula, at the center of this particular blast, cells are called inner cell mass cells. These cells are basically give rise to each and every cell type of our body. Okay, so then we know that over a period of time, by means of a cell proliferation and then differentiation, gradually the single cell zygote develops into adult organism, and then adult organism again from the germ cells again it produces the gametes. Thus the cycle will be completed. So as you can see that the formation of a zygote is very, very important step. That is actually formed by the union of haploid nucleus from oocyte and then haploid genome from spore, thus become diploid nucleus. That means at this particular level, the single cell zygote has the every information is encoded for the formation of a particular organism. If it is a human zygote, it develops into human. If it is a mouse zygote, it develops into mouse. But one of the very, very fundamental question is that how come that single cell that we all started has every capacity to give rise to such a complex organism? Next slide, please. Yeah. So as I showed you that, so from starting from this particular inner cell mass cells, so which basically these are the cell types which can give rise to any cell type of our body. They are called pluripotent cell or totipotent cells. So because that they can give rise to more than one different kinds of cell type, they are called stem cells. So these cells which are present at the embryo, they are called embryonic stem cells. Embryonic stem cells, to start with, they are pluripotent. That means they can give rise to any cell type of our body. So it is not that directly stem cells can give rise to every differentiated cell. As you can see, there exists a hierarchy. So starting from pluripotent stem cells, you will have a multipotent progenitor cell. For example, ectodermal progenitor cell, mesodermal progenitor cell, and then endodermal progenitor cell. Then eventually these progenitor cells give rise to various cell types of our body. For example, ectodermal progenitor cell can give rise to neurons, glial cell, and epithelial cells. All the cells that are derived from ectoderm, mesoderm, and then endoderm. Next slide, please. Yeah, then we know that, see, at this early embryonic development, all those cell types which are specified eventually give rise to various cell lineages. For example, ectoderm, as you can see, it is mentioned here, different cell types of our body is derived from ectodermal progenitor cell. And then mesoderm, as you can see, notochord, skeletal system, all these things are derived from mesodermal progenitor cell. And then endoderm finally give rise to multiple lineages, as it mentioned here. That means from a single cell zygote, how that one can generate such a complex multiple cell types of our body. For example, in human body is really made up of approximately 175 different kinds of cell types. And then if you look into all these cell types, next slide, please. See, if you look into all these cell types, they actually once upon a time derived from a same common progenitor cell. So here, for example, embryonic stem cell. From this embryonic stem cell, you see that multiple cell types are generated. If you look into all these differentiated somatic cells of our body, they have a identical genome that is basically derived from single cell zygote. Single cell zygote, by means of mitotic cell division, give rise to multiple cell types. That means genome of each and every cell of our body are identical. The genome is nothing but it is the genetic material, that is deoxyribonucleic acid, which is there in the cell, in the form of a cell nucleus in our body. Next slide, please. See, this particular slide show you that, so we are made up of multiple cell types. Each and every cell, as you can see, you see that there is a our genetic material is present in the form of a spherical nucleus. So here in this particular photograph, you can see the nucleus in the form of a blue color because this particular cell is stained with a dye called DAPI, when you observe these DAPI stained nuclei under fluorescence microscope, you can see them as a blue color. You can see that some cells which are in interface stage, you see that entire nucleus is fluorescing with blue color, indicating that DNA is present exclusively in the nucleus. And we know that this particular cells undergoes mitotic cell division. So when these genome during cell division, you see that they condense, they become appear like a chromosomes at particular stage of cell cycle. We know that during metaphase, the chromosomes can be seen, but same chromosomes are not seen during interphase. They seems to be present throughout the nucleus in the form of a very diffused pattern. So at the same time, we know that, for example, if humans, we are every cell of our body, our human cells consists of 46 chromosomes. That means all 46 chromosomes are present in each and every cell. Next slide, please. So as I said, each and every cell has an identical genome. And then if you look into this particular gene, we know that our genome is a deoxyribonucleic acid. At the very fundamental level, the DNA is not naked in the nucleus. It's always in association with a group of proteins called histones, forms this kind of nucleosome-like structure, which is called beads on string. When you observe these kind of structures under electron microscope, it looks like that. The picture in the left side shows the appearance of a beads on string-like form, which actually nothing but DNA and then nucleosomes together forms a chromatin. So DNA and then and histone proteins form nucleosomes. This nucleosomal DNA is called chromatin. So chromatin exists as a very loosely arranged wheel-like string-like structure, as well as very higher orderly folded, very compact chromatin as well. Next slide, please. Yeah. So further go deep into the individual nucleus. As you can see, this is an electron micrograph picture of a typical human cell nucleus. As you can see clearly that within this particular nucleus, there are two different regions. One is electron dense material and then electron poor regions. Electron dense region is, we know it is called heterochromatin, and then electron poor material is called euchromatin. Euchromatin, and we know that euchromatin is nothing but it is the region with genes are highly active. Heterochromatin is the region where the genes are highly silent. So that means all genes are present in all the cell types, but all genes are not transcribed in each and every in all the cells. So every cell on average transcribes only subset of genes. For example, if in humans, our genome codes for nearly 30,000 genes. But all these 30,000 genes are not transcribed in each and every cell. So, for example, brain cells transcribe some 5,000 genes, and then liver cell transcribes some around maybe 5 to 6,000 genes. But if you try to see the genes which are transcribed and then translated in brain and then liver, they are different. Okay. So in that context, if you look into this, our genome in the nucleus, so it exists as a heterochromatin as well as the euchromatin. Euchromatin, I can see it's very loosely arranged chromatin. A heterochromatin, highly compacted chromatin. And these particular diffuse pattern of chromatin that you can see in the interface can be seen in the form of a chromosomes during a particular stage of cell cycle. So during interphase, you don't find chromosomes. You find only the very diffused chromatin. Next slide, please. And then other very important thing that we need to understand is that, so now we have seen in previous slide the chromosomal structure. So chromosomes that I showed you in previous slide looks like a X chromosome, looks like a X-shaped chromosomes, right? So if it is very important for us to understand, it is important for us to understand how the DNA molecules are actually arranged within this X-shaped chromosome. So in this slide, as you can see, a DNA molecule before undergoing duplication. I think I see some voices coming up from NRC Tanuku. So can you please mute yourself, please? Yeah, thank you. So as you can see, you have a DNA molecule. So DNA is a right-handed helix. So during replication, what happens is that these original DNA strands are unwound and then newly synthesized DNA being synthesized by semi-conservative replication process. So as a result, single DNA molecule after replication process results in the two DNA molecules. You can see in the two DNA molecules, one is older strand, other is a newly synthesized strand in the orange color. So if you look at the same kind of structure in the chromosomes, the X-shaped chromosomes has, you see that one of the sister chromatids has one older DNA strand, and then one newly synthesized DNA strand. So this kind of understanding is very, very important for us to really understand how that during mitosis, the genome is duplicated and then they're equally segregated between the daughter cells. And then these X-shaped chromosomes during metaphase, we know that during anaphase, they each chromatid move in opposite to poles. Thus, they will become a single rod-shaped chromosomes. Now we can see that chromosomes exist as X-shaped chromosomes because they are duplicated. Then after separation, they appear like a rod-shaped chromosome. That is what exactly you can see during anaphase and then telophase. Next slide, please. And then just to give you very basic information about genes. So our genomes are having genes, and then genes are basically characterized by a promoter just upstream of this particular gene, and then each and every gene starts with exon and then followed by introns. Because in eukaryotes, our genes are called split genes because we have exons and introns. So when these genes are transcribed by RNA polymerase II enzyme, so from DNA, it is converted into RNA by process of transcription. This RNA that is initially formed, it has both exonic component as well as the intronic component. And then later within the nucleus, when this primary transcript is generated, it has both exonic sequences as well as the intronic sequences. Then by process of splicing, all these intronic sequences, such as which is mentioned, which is labeled with the red color, they will be specifically eliminated. Then all exons which are in blue color, they are specially added. So thus you will have a matured messenger RNA that comes out of the nucleus and into the cytoplasm, and then bound with the ribosomes for the translation process, giving rise to proteins. So from gene, this is the central dogma of life. From genes, process of transcription give rise to RNA. RNA by process of splicing give rise to mature messenger RNA. Mature messenger RNA goes to the cytoplasm and then bound with the ribosomes, and then by process of translation, you will have a respective proteins. So these proteins decide the functions for a given cell type. Okay, so these genes, so as I said, in human genome we have 30,000 genes. All these 30,000 genes are not transcribed. Only few genes are transcribed in a cell type specific manner. So when genes are transcribed, these promoters are very, very important for RNA polymerase to bind and then for transcriptional regulation. So at the same time, there are multiple sequences which are present away from the gene body, which are very important for transcription status of a given gene. These are called regulatory elements. So one of such regulatory elements are enhancers. Enhancers are the sequences which are present away from the gene body, but during transcriptional activation, as you can see in the right side of the figure, there is a gene, and then there is enhancer which is labeled in the red circle. So when this gene is active, you see that distally placed enhancer come near to the gene promoter, thus gene can be transcriptionally active. So in absence of this physical proximity of enhancers, genes are not usually transcribed. Next slide, please. So at the same time, one of the very important questions is that, see, every cell has all the genes, but why that only few genes are transcribed in liver? For example, liver transcribes liver specific genes. But how a liver cell knows that I am a liver, I need to transcribe liver specific genes? For that matter, neuron transcribes only neuronal specific genes. But how a neuron knows that I am a neuron, I need to transcribe neuronal specific genes? So research from the last decade has shown that there are some kind of hidden signatures over the genes in a cell type specific manner. So any gene that is off in any cell type, if you try to look into the DNA, DNA is heavily methylated. The cytosine moiety of that particular gene, where it is transcriptionally off, you see that that particular C is heavily methylated. Okay, so whereas the active genes, if you see, those regions are cytosines are there is no evidence of methylation. So the active genes are devoid of DNA methylation. Okay, and further, you see this the DNA along with histone proteins forms in nucleosomes. If you try to look into all repressed genes in a cell type specific manner, they have a regularly placed nucleosomes, whereas transcriptionally active genes has irregularly placed nucleosomes. Further, these histone proteins undergoes post-translational modifications and which are specific for active genes and then silent gene. That means someone has already written in the form of some kind of code over in the form of histone modifications. So for example, histone H3 K9 lysine 9 position undergoes trimethylation is a hallmark for all repressed genes. And then other modifications such as histone H3 K4 trimethylation is a hallmark for all active genes. In general, all active genes have highly acetylated histones, and then all the repressed genes histones are heavily deacetylated. Further, at the higher order chromatin level, in a cell, when the gene is transcriptionally active, it is in the form of a chromatin fiber, which is highly loose chromatin fiber, whereas all repressed genes are having this kind of a higher order folded compact chromatin structure. That means someone has written in the form of some kind of code that will tell you that you are a so-and-so gene in a so-and-so cell type. You can be transcriptionally on or you can transcriptionally off. That is decided by DNA methylation, nucleosome positioning, histone modification, and then chromatin higher order compaction status. For example, insulin gene is present in pancreatic cells only, right? Insulin protein can be seen only in the pancreatic cell. That means insulin gene is transcribed only in the pancreatic cells, but neither our brain cell or liver cell, they don't transcribe insulin gene. But how a insulin gene knows that it has to transcribe and translate only in pancreatic cells only. So when scientists looked at those cells, insulin gene in pancreatic cells has all these active histone modification. Their gene bodies divide of DNA methylation. They have irregularly placed nucleosomes. They have active histone modification, and their chromatin is loosely arranged. Same insulin gene, if you look into liver cells, their DNA is heavily methylated, and then nucleosomes are regularly placed, and then they have a silent histone modifications, and then their chromatin is highly compact. See, someone has already designed genome in a cell type specific manner that will decide whether gene is going to be transcriptionally active or not. Next slide, please. Next slide, please. Yeah. Then see, I showed you the chromosomes, right? They can be seen as X-shaped structures or rod-shaped chromosomes during anaphase telophase. But in interphase, what is the status of these so-called rod-shaped chromosomes? Are they still seen as a chromosomes? No. So then scientists use a technique called chromosome page. So you can locate a particular chromosome-specific DNA within the interphase cell nucleus by using a technology called chromosome paint. So here in this example that I gave you, here scientists try to locate chromosome 1 and then chromosome 2. Chromosome 1 they label with the green color, chromosome 2 they label with red color fluorescence. When they hybridize onto the interphase cell nucleus and then try to locate the distribution of this chromosome 1 and chromosome 2 specific DNA by labeling with the different fluorophores, they see this kind of picture. They see that two bigger chromosome 1 signals and two bigger chromosome 2 signals in the form of a red color. And see, always it is not appearing like a chromosome, right? So it is a DNA belongs to particular chromosome, but occupies a particular territory within the three-dimensional space of the nucleus. These are called chromosome territories. These are called chromosome territories. And then here one can find chromosome 1 seen in two different locations in the form of a chromosome territories. We are seeing two locations because we are, as you know that we are diploid animals. If you take any chromosome, we have a mother component as well as the father component. That's why whenever you're trying to decrypt a particular chromosome, you see them as a two different locations. And then here you can see that chromosome 1 occupies a unique territory, chromosome 2 also occupies a unique territory. Okay, so that's how scientists learned that during interphase cell nucleus, nuclear genome chromosomes, chromosomal chromatin occupy discrete constrained locations. They are called chromosome territories. Next slide, please. See, this is the actual pictures. So then scientists were very much interested to understand. So if a chromosomes seen as a chromosome territories, and then simultaneously they label three different chromosomes, one chromosome with green color, one chromosome with red color, other chromosome with blue color, simultaneously try to detect three chromosome territories in a single nucleus. They noticed that some of the chromosomes are neighbors. For example, this here, red chromosomes are green color chromosome is near to blue color chromosome. So then they thought, then they see that some chromosomes are always seen together in a cell type specific manner. So then they really wanted to understand whether that a particular chromosome territory position within the nucleus is specific to each and every cell type. So next slide, please. Next slide, please. Yeah. So then they wanted to know the intranuclear distribution of all the chromosome territories. So they used a technique called SCI, spectral karyotyping, where each and every chromosomal DNA now labeled with a different fluorophore. Okay, then they detected all the chromosome territories in the interphase cell nucleus. As you can see in the figure, this sphere nucleus. You see that every chromosome has a chromosome territory, and you see that for example, red chromosome and then green chromosomes are together, and they're away from the chromosome blue chromosome. Then from these kind of spectral karyotyping, scientists have learned that although all cells are having identical genomes, but the way each and every chromosomal specific DNA is arranged three-dimensionally is unique for each and every cell type. That was one of the breakthrough finding that scientists have identified that although genome is same, DNA sequence is same in every cell, but that the way each and every chromosomal DNA is placed within the three-dimensional space of nucleus is unique for each and every cell type. We are aware of the fact that each and every cell type has a unique transcription program. But now we are coming to know that each and every cell type has a unique three-dimensional arrangement of chromosome territories and then their neighborhood. Next slide, please. Yeah, this cartoon show you how that so-called mitotic chromosomes, that what you see during anaphase, when they go to daughter cell state, they see that they gradually decompact. See 200-fold, 1,000-fold. When it comes to interphase state, they unfold by 400,000 times, but still they occupy a unique territory. So likewise, each and every chromosome occupies a unique territory, and then these kind of territory and then neighborhood is cell type specific. That means chromosomes are readily positioned and their organization is cell type specific and is linked with the differential gene expression in a cell type specific manner. Next slide, please. Yeah, then question comes how that scientists could able to identify that although all cells are having identical genomes, but we have different configurations for each and every chromosomal DNA in a cell type specific manner, which are functionally linked with cell type specific transcription program. So there are some kind of tools which are developed over some 10, 12 years ago that has actually revolutionized the way people try to understand the how genes are physically located in the nucleus, how their location is important for cell type specific transcription program. So these techniques are, I just put them here. Like these techniques are chromosome conformation capture. In short, it is called 3C technique. By using this technique, one can identify a particular gene and then its regulatory elements such as enhancers, silencers, and all other regulatory elements. So this is called chromosome conformation capture. So one-to-one interaction. That means you have a gene promoter and one can identify its respective regulatory elements by performing 3C. And then from this technique, we also have a technique called 4C. It's called 3C on chip 4C, 4C or circular 3C is called 4C technology. By using this technique, one can identify a particular gene and its interacting genes within the nucleus. As you can see here, there is a gene in chromosome number 1 and it is interacting with multiple genes across different chromosomes in this particular cell type. So by using 4C technology, one can identify in unbiased fashion the interaction between a particular gene and its other genes within the nucleus. I will tell you the significance of gene-gene interactions in the nucleus in the coming slides. And then the other technique called HiC. By using this technique, one can identify the entire genome, how that each and every gene is placed, how the chromosomes are placed, how the chromosomes are positioned within the spherical nucleus, and then how that euchromatin, heterochromatin are organized within the chromosome, and then how that individual genes are organized. All this information can be deduced from a very powerful technology called HiC, which is linked with next generation sequencing. By using this technology, one can decipher the entire three-dimensional organization of a particular genome. We know that three-dimensional organization of genome is unique for each and every cell type. So this technology has revolutionized the way now with current day understanding about how that genes and genomes are organized in the nucleus. Next slide, please. Next slide basically tells you how actually 3C is performed. 3C is a very simple molecular biology technology in which cells are basically, to start with, you take a cells and then you cross-link with any kind of a cross-linker such as formaldehyde so that all DNA protein contacts are cross-linked. And then these are subjected to recession enzyme digestion. Thus, it specifically cuts the DNA. And then this is followed by ligation with the T4 DNA ligase, resulting in the ligation of any two closely placed DNA molecules which are having same kind of enzyme sites. So now, for example, think that you have a promoter, you have an enhancer, they are separated by 1 MB, but they are brought together by protein-protein interactions during gene activation. Because of this procedure, they are ligated. Now we have a primer from enhancer, primer from promoter. If you just do PCR, you will see the PCR amplification. So presence or absence of a PCR amplicon tells you that whether they are interacting within the nucleus or not. So likewise, scientists have identified the enhancer promoter communications. Next slide, please. So by using this 3C, next slide, please. So by using this 3C, as you can see in this example, a gene-specific enhancer can be identified. And at the same time, in yeast, chromosome 3 and then chromosome 4, all centromeres of different chromosomes are come together. Even one can demonstrate the proximity of centromeres of chromosomes in yeast by using 3C technology. Next slide, please. The next slide show you. I'm going to show you the procedure for 4C. 4C, as I said, for example, here is the single cell nucleus where you have a particular gene. When the genes are transcribed, it interacts with the multiple genes. So all those multiple genes to which a particular gene is interacting can be identified by using this kind of procedure, which is basically biotonated nucleotide, which is corresponding to your gene of interest, that can be used for a primer extension, then followed by using the streptavidin coated paramagnetic bits, you can pull down this particular DNA fragments. Thus, then following the sequencing will tell you which are the genes which are in close proximity with your gene of your interest. Next slide, please. Next slide show you how the technique called HiC. Yeah, next slide, please. Yeah, this is the HiC technology. It is similar to that of a 3C technology, where the two cells are cross-linked with formaldehyde, they are digested with HIN3, then leaves behind this kind of cohesivants, and then they are end-filled with biotonated nucleotides, and then they are subjected to ligation. And then you see that now junctions which are formed, these are two remote DNA elements which happens to be proximal. They are ligated because they are proximal. And then all these ligated junctions having biotin, later by purifying the DNA, and and then enriching specially these junctions which are having biotin by using paramagnetic beads, streptavidin coated paramagnetic beads. One can isolate all these millions of junctions and they can be subjected to sequencing. And then next slide, please. After sequencing, one can derive the three-dimensional organization of entire genomes. So this is actually sequencing data. Sequencing, the millions of pairwise sequencing data can be used to deduce whole genome contact matrix. And then, as you can see, this is a mouse chromosome. Mouse 20 chromosomes can be seen. From these whole genome contact matrix, one can deduce chromosome territory positioning and then their neighborhood. Further, same data can be used to study the organization of individual chromosome territories, which will tell you the organization of chromosome territories. And then further, you can go deep into these particular domains, and further it will tell you this kind of it will show you this kind of triangle shaped structures. They're nothing but these are DNA loops. So genes at the chromatin level, at the DNA level, at the end, all our genes are in the form of a loops that can be seen in the form of a domain. So that means by doing single high-C technology, one can identify chromosome territories and their neighborhood. Within chromosome territories, you can identify the euchromatin, heterochromatin, which are the active and then repressed genes. And then at even much at higher resolution, individual genes, how actually they are organized, also can be provided by this high-C technology. Next slide, please. By using this high-C technology, now we know that. So I showed you how the chromosome territories looks in the interface cell nucleus. So within this interface chromosome territories, you will have a compartment A, compartment B. Compartment A is euchromatin, which are transcriptionally active, and then heterochromatin, which is transcriptionally repressed. And then if you go deep down, you will have this kind of ball-like structure with the self-interacting chromatin domains called topologically associated domains. So all gene regulation in eukaryotes happen in this tiny ball-like self-interacting domains called topologically associated domains. If you see here one ball that may have, if you go down, it has a lot of loops where genes are interacting with the enhancer, genes are interacting with the silencers, or you also have a base of the loops. So you see that structure what you see at the individual genes in the form of loops are hierarchically organized over the chromosome territories, and then even chromosomes are hierarchically organized in a cell type specific manner. All this data derived from these important molecular tools that I described, 3C, 4C, and then IC. Next slide, please. So this is how example. See here, in one cell type, you have 4 loops. So for example, as you can see here, seen at the bottom, 4 loops, and then loop 1, loop 2A, loop 2B, and then loop 3. And then if you see that particular organization form of triangles, see in one particular loop, you have a gene and then enhancer. In another loop, you have a gene, but enhancer is not available. Because the enhancer is not available, this gene is not transcribed. But in other triangle, you have a gene and then enhancer. Because they are in the same loop, they can be transcriptionally active. You see at the bottom of this particular loop, see in one loop you have a gene and enhancer, that gene is transcriptionally active. In other loop you have a gene but there is no enhancer, that is repressed. But because of some kind of mutations, okay, these particular borders are erased. As a result, now instead of forming a 2 loops, it forms a single loop. Now you see that in a single loop you have one gene which is regularly transcriptionally active. Now the other gene which happens to be the oncogene, which otherwise repressed because of this changes in higher order chromatin organization. Now enhancers can access oncogene, thus normal cell can become oncogenic in nature. That means structure of a gene is very, very important for its transcriptional activity. Structure keeps genes in silent condition. Similarly, same structures can keep genes in active condition. Any perturbations in this chromatin organization leads to dysregulation of gene expression, which are reminiscent of a diseased state. Sorry. Yes, next slide, please. See, now you see that, so every cell type has a unique genome organization. So, for example, liver cell give rise to liver daughter cells. So what about the genome architecture? So genome architecture of a parental liver cell where you see that you have euchromatin marks and heterochromatin marks. When this cell divides, when these chromosomes become chromosome territories or chromosomes, you see that both heterochromatin and euchromatin marks in the form of orange and blue colors are retained over the mitotic chromosomes. And even after anaphase and segregation, you see that when the two daughter cells are formed, they have a signatures of in the form of histone modification, euchromatin and heterochromatin marks. Thus, when they form chromosome territories in the daughter cell, again you see that all heterochromatin goes to the nuclear periphery, all euchromatin can be seen in the nuclear interior. Thus, all heterochromatin is repressed, all euchromatin is transcriptionally active. Thus, the transcription memory of a parental cell are recaptured or memorized through mitotic cell division. Thus, when the cells are formed out of metastasis, they also remember the parental cell specific gene expression pattern because they memorize the nuclear architecture, genome architecture, and then histone modifications. Next slide, please. Yes. See, right now, I told you that chromosomes exist as a chromosome territories, and these chromosome territories are unique for each and every cell type. Each and every chromosome territory has a euchromatin and heterochromatin part. Heterochromatin part of each and every chromosome territory is seen in the nuclear periphery. Euchromatin part is seen in the nuclear interior. So then, and this structure is maintained during mitosis. But we know that during cell differentiation, for example, you have an embryonic stem cell. They are differentiated to become a neural progenitor cell and then finally differentiated into astrocytes, for example. During this differentiation process, now we know that clearly not the same genes are present in embryonic stem cells and neural progenitor cells and then astrocytes. These three cell types transcribe unique set of genes, but they are all having identical genomes. During this transition from embryonic stem cell state to neural progenitor, for example, you see that all embryonic stem cell specific genes, which are in the form of a loop organization, they become part of the heterochromatin and they are repressed. Whereas the neuronal specific genes in embryonic stem cells are part of the nuclear lamina, and then in neural progenitors, you see that they loop away from the nuclear periphery for transcriptional activation. That means genes dynamically switch between heterochromatin to euchromatin, euchromatin to heterochromatin, or other put it in other way around, the nuclear periphery to interior, interior to nuclear periphery, depending upon the cell type. That is what exactly dictates transcription status of a given cell type. So genes dynamically shift from euchromatin to heterochromatin, heterochromatin into euchromatin as a matter of cell differentiation process. Next slide, please. Okay, so from these till now, I discussed with you the structural organization of our genome, which we learned that it is cell type specific, right? So at the same time, parallelly, there are a lot of nuclear functions, such as one of the key nuclear functions is transcription, replication, recombination, splicing. So these are all nuclear processes. So look into this important nuclear process of transcription that is performed by enzyme called RNA polymerase II. If you try to locate a RNA polymerase II enzyme in the nucleus, it appears like this. You see here the red fluorescence. You have a lot of nuclear foci. It's called a red focus, which fills the entire nucleus. So for that matter, if you take any nuclear protein, it's always seen in the form of this kind of a nuclear domain. They are called nuclear domains or nuclear foci or nuclear domains. Next slide, please. So research from the last decade were aimed at understanding what exactly these nuclear foci are. So before going to that particular nuclear foci, what exactly they are, there are some kind of fundamental things that we need to understand. See, soon after the sequencing of the entire human genome, scientists learned that our genome has 30,000 genes. If you try to see the distribution of these particular genes over all these 23 chromosomes, it was learned that these genes are not arranged randomly over the linear sequence. Instead, they are clustered at the primary sequence level. Not only clustered, but they are clustered and co-expressed in a cell type specific manner. So these kind of regions, which are clustered genes, which are co-transcribed in a cell type specific manner, they are called regions of increased gene expression. In short, they are called ridges. Next slide, please. Hello. Okay. So, see, I showed you that transcription enzymes, RNA polymerase to polymerase, exist in the form of nuclear foci, right? So, for example, if you try to see, so one end we learned that in a given cell type, approximately 5 to 6,000 genes are transcribed. But every gene requires RNA polymerase 2. But a cell transcribes 5 to 6,000 genes. So if I try to locate RNA polymerase 2, then I would expect to I expect to see this kind of grainy pattern, because each and every grainy appearance is nothing but is a single RNA polymerase 2 actively engaged in translation of a given gene. But in reality, when scientists have done a technique called immunofluorescence to detect RNA polymerase 2 enzyme in the nucleus, they see this kind of pattern, observed pattern. It forms some few large nuclear foci. Okay, then scientists were puzzled. What are these nuclear foci? Are they storage sites for RNA transcription, for RNA polymerases? Next slide, please. So then scientists were investigated why that nuclear process transcription is compartmentalized. And then they see, this is the exactly red color showing the location of RNA polymerase 2 engineering within a single nucleus. And then when you try to detect transcripts, for example, here they detected transcript of globin RNA in the form of a green color. You see that transcript is always seen at the RNA polymerase 2 red focus. So these focus RNA polymerase 2 enriched nuclear foci are nothing but these are the regions. Okay, for example, 1,000-fold higher concentration of active RNA polymerase 2. Okay, per nucleus you find that 200 to 400 foci per cell. But in a given cell, we know that there are 4,000 genes are transcribed in terms of alleles, 8,000 express alleles. But this has given the indication that these RNA polymerase 2 enriched centers are not storage site. This active transcription is going on. So how many genes are transcribed in a single foci? Next slide, please. So now in this experiment, scientists wanted to know, see, if for example, there is in mouse, there are chromosome number 7. There are 5 genes, for example, HBB, red color, ERAF, Euros, IGF2, KCNQ1, they are labeled with green color. So in first experiment, what they have done is that they have taken HBB probe and then they have taken ERAF probe, as you can see in the panel, HBB and ERAF. So when they try to detect these two genes, these two are transcribed in these particular cells. They see that in 42% of the cells, you see that both red color and green color are together, giving rise to yellow fluorescence. And then next you see, then next experiment they have done, same HBB gene red color, and then they have taken Euros. Then you see that again 41% they are colocalized. And then IGF2, they colocalize 55% of the cells. And then KCNQ1, they are colocalized with 60% genes. But on the linear scale, they are separated by megabases away. But when you try to do in reality within the nucleus, they are seen together, suggesting that these distantly placed genes above the chromosomes, when they undergo transcription, they come together to a common center of RNA polymerase in this center for a transcriptional activation. Next slide, please. And here is an example to show that in one particular cell type, there are 2 genes, TFF1 and then GRB1, these 2 genes. You see TF1 is labeled with green color. They are away from each other. You see two green spots, 2 red spots. But when you give estrogen, you see that now they come together. So before estrogen, they are not transcribed. After giving estrogen, they are transcribed. That is correlated with these genes coming together. That means genes are kissing each other in the same nucleus. That means they are coming together. Next slide, please. So these experiments unequivocally demonstrated that in eukaryotic system, when the genes are transcribed, they are not transcribed alone. They are transcribed in group. As you can see here, so you have a single cell with chromosome territories and with RNA polymerase 2 centers, which is magnified here. You see that genes from same chromosome are different chromosomes, three-dimensionally cluster and forms a dedicated centers for transcription. These are called transcription factories. or transcriptional condensates. It's like a, for example, in a hostel, we have a common kitchen, but we don't have a kitchen in each and every room. Why we don't have a kitchen in each and every room? It is more economical and robustness wise more economical in the self point of view. Thus, multiple genes which requires to be transcribed, they come together in a centers where all the transcription machinery are available, and they go transcribe all together in a same cell. These kind of structures are called transcription factories and then transcriptional condensates. So it is not only for active genes. Next slide, please. Even same phenomena can happen in repressed genes as well. Okay, this is just to show you that during transcription, like textbook, our textbook tells you that whenever you heard about transcription, you imagine that DNA is stable, RNA polymerase II binds to DNA and then moves along the DNA. So that is what exactly our perspective is. As it moves along the DNA, it produces corresponding RNA. But in reality, in the nucleus, now what is clear is that RNA polymerase II enzymes are stable, whereas the DNA is reeled through. During this, when it's reeled through this DNA transcription center, you will have a corresponding RNA produced. Just imagine you have this kind of multiple genes are coming together at the centers of RNA polymerase II, and then they are co-transcribed. Next slide, please. Yeah, as I said, not only active genes, even silent genes within the nucleus. So in this example, for example, you see that there is a gene in Drosophila called ARTP, labeled with red color. There is another thing called ADB, labeled with pink color. But these two genes are repressed in the head region of Drosophila. As you can see here, you will find a red color and or pink color together, and then there is a protein called polycomb group of protein, which is a protein response for gene repression, which also form this kind of nuclear foci, which is a green in color. So these genes, when they are transcriptionally off, they are together. The merged image shows you that both gene 1, gene 2, when they are repressed, they are within the nuclear foci of a repressive genes. But in the posterior part of this particular Drosophila, one gene is on, that ARTDP red gene is on. When this gene is on, as you can see, this foci is away from the polycomb enriched nuclear center. That means not only for gene activation, even for gene repression, genes cluster three-dimensionally for active repression. Next slide, please. Yes, this is just a cartoon show that even for repression, genes from same chromosome or different chromosome can come together for coordinated repression of genes. Next slide, please. Yeah, so this cartoon should give you information about. You see that, see, we learned about electron microscopy, electron dense heterochromatic nuclear periphery, and you see within the chromosome territory, as you can see here, all these chromosome territories with the repressive genes which are placed intermittently, they all are clustered and they are pushed over the nuclear periphery. All the active chromatin or active genes in the blue in color, they are seen in the nuclear interior. Next slide, please. Yes. See now, we learned about chromosome territories, and then chromosome territory positioning is unique for each and every cell type. But during stem cell differentiation, that means from these stem cells basically give rise to every differentiated cell. When you look into these stem cells, chromosome territories are highly random. They do not have any defined location. They are highly mobile within the embryonic stem cell nucleus. But as they differentiate, they form extensive interchromosomal contact. Thus, each and every cell type has a unique three-dimensional arrangement of chromosome territories. Next slide, please. So, as I said, these kind of so genes from same chromosome territory or different chromosome territory, whatever they, along with RNA polymerase II whole enzyme complex, they form this kind of membraneless higher order structure called transcription factories by a phenomenon called liquid-liquid phase separation. So liquid phase separation is the biochemical phenomenon. It is just like if you take a glass of water and if you add a drop of oil in it and then shake it, it forms emulsion. Again, you keep this particular glass in a standing position for a couple of minutes. All these dispersed oil droplets, they fuse together, finally forms a single oil droplet. So both are liquid, but their phases are separated. Similarly, within the nucleus, the RNA polymerase II and then associated genes, all these complexes forms this kind of a transient dynamic structures called structures by a phenomenon called liquid-liquid phase separation, which are reminiscent of a transcriptional activity or repression in eukaryotic cells. Next slide, please. Yeah. So I think with this, I covered both structural organization of genome and then how that functions are orchestrated within this particular structure. Next slide, please. Yeah. See, other important thing that we learned about our DNA, right? If you look into DNA of all these 23 chromosomes you put together, it measures around 1 meter. So how that 1 meter length DNA, since we are deployed, this 2 meter length DNA are packed in such a tiny volume. If you have a 2 meter length DNA thread, if I ask you to put it in the smallest possible volume, we can put it in this particular way. Next slide, please. So in this way, you can put it them in the smallest possible volume. But if you really wanted to access any region, you cannot access the particular region. So that certainly our DNA is not organized in the particular fashion. So then there are, in polymer physics, there are two ways one can arrange a particular polymer in a fixed value. You see, one is Pinot-Car value pathway, other is Hamilton pathway. As you can see, in a Pinot-Car, so this is the start point, and the red is the start point, and the blue is the end point. But if you look into Pinot-Car pathway, the start point of the polymer is always near to its nearby regions, right? There is Hamilton pathway, it is randomly arranged. Although both of them are fixed in the same value, but this Pinot-Car pathway is not free. It never forms knots. The Hamilton pathway, it is prone for knots. But our DNA polymer, how exactly it is actually arranged? Next slide, please. So through this high-speed technology, scientists have learned that chromatin within the nucleus, okay, is organized into knot-free fractal geometric polymer configuration. You see, now it follows the Pinot-Car pathway. That's why you see specific chromosome territories. So what is this fractal? So next slide, please. So fractals, I think you must have heard about these fractals. Fractals are everywhere in our nucleus. First of all, our universe itself is a fractal. Okay, the next slide, please. You see, so fractal is nothing but like a self-similarity. That means the structure, for example, around solar system, all our planets are rotating. Again, solar system itself is rotating against something else. Again, that itself is rotating against something else. That means the structure, what you see at the very microscopic level, the same structure repeated multiple times. This is called fractal geometry. Similarly, within the nucleus, at the microscopic level, you have a topological set of domains, then you have euchromatic heterochromatic domains, and then chromosome territories, and then they are arranged in a cell type specific manner. So genome in our nucleus are fractal geometrically organized. Thus, it will never form knots. Next slide, please. So this information is very, very important. Otherwise, people think that we have a lot of DNA molecules in the nucleus. Why that they never form knots? They never form knots because they are organized in Pinot-Car pathway, mimicking the fractal geometric configuration. So this particular fractal geometric organization will explain the heterochromatin and euchromatin. Beauty with this particular model is that every region, any region that you are interested in, it can be easily accessible to any kind of functions. Next slide, please. Yeah, next slide. So this is the same thing which I tried to show, that fractal geometry. Okay. Other very, very important thing that we should be aware that, see, when the genes are active, you see there is a technique called DNA. One can perform DNA fish technology to locate the gene of your interest. So here, red color is the location of a gene, and then green color is the location of its transcripts. See, active genes, this can be detected by DNA, RNA, FISH, fluorescence in situ hybridization. When the genes are active, they are seen at the nuclear interior because both the red dots are interior, and then you perform RNA FISH, both are given in the green color, suggesting that genes are active and they are present in the nuclear interior. We have a genes, for example, same thing if you try to do for a silent gene, by DNA FISH you can detect them, but they are seen at the nuclear periphery. And you also perform RNA FISH, but you don't detect green color because they are not transcribed. Similarly, we have a group of genes called imprinted genes. In the same nucleus, out of the 2 copies, 1 copy derived from mother is active, other copy derived from father are inactive. So in such kind of if you take such kind of genes, so when you locate the gene, one gene is seen at the nuclear periphery, other gene allele is seen at the nuclear interior. When you perform RNA FISH, RNA transcription technology, RNA FISH, you see that the allele that is there in the nuclear interior, you can see the green color, suggesting that it is transcribed. Allele at the nuclear periphery do not provide the green color, then it tells that the gene is not transcribed. That means there is a relationship between gene positioning and then transcription status. The gene is present in the nuclear interior, it is transcriptionally active. It is present at the nuclear periphery, it is transcriptionally repressed. Next slide, please. Similarly, other important topic that I'm trying to bring you that. So then question comes, who decides the gene position? Next slide, please. Next slide, please. Yeah. See, in a cell, if you have a parental cell, when it gets a signal that it has to undergo mitosis, genome has to be duplicated. When the genome is during duplication process, not the entire genome is replicated at a time. The genes which are in euchromatin, they tend to replicate early because they're already freely available for DNA polymerase I enzyme to recognize. But heterochromatin is highly compact. So during the replication, heterochromatin has to be decompacted. Thus now DNA polymerase can access these particular regions. Thus they can be replicated. That means during replication process, euchromatin replicates early, heterochromatin replicates late. That means replication timing will act like a epigenetic memory for transcriptional activity. All those genes are replicated early, they are transcriptionally active, and then in the daughter cells, all those genes which replicate late, they have a transcriptional memory of repression. Accordingly, in the daughter cells, they are repressed. Next slide, please. Next slide, please. Yeah. See, I think this is the last slide. See, from this you can understand that just DNA sequence per se will not tell you which gene is going to be active or repressed. So it is gene transcription is controlled at multiple epigenetic levels. For example, pre-transcription level itself, gene transcription status is decided. That is, in that context, DNA methylation. If it's methylated, it is most likely is repressed. Replication timing. If it replicates early, this gene is most likely to be transcriptionally active. If it replicates late, gene is repressed. And then chromatin accessibility. If it's highly accessible, then it tends to be accessible to transcription message. It can be transcriptionally active. If it is not accessible, then it most likely it is repressed. Chromatin condensation, chromatin supercalling, three-dimensional chromatin organization. As I mentioned, that three-dimensional organization of genes will decide the transcription status. Further, genes are also regulated at the transcription level by promoter proximal pausing, presence or absence of a few cell type specific transcription factors, histone modifications, chromatin remodeling also decide which genes are going to be transcriptionally active. And then genes also controlled by post-transcription level, such as alternative splicing, microRNAs, long non-coding RNAs, circular RNAs, even RNAs are undergoes modifications called epitranscriptomics, and then RNA binding proteins, mRNA stability and degradation. All these things also play an important role in epigenetic control of gene expression. Next slide, please. Further, genes are also controlled by translation level, at the level of specialized ribosomes, during initiation, elongation, and then termination. And then there are a group of proteins called microproteins. They also control the gene expression at the post translation level. And gene expression also controlled by post translation level, that is at the level of protein post translation modifications, protein stability, and protein degradation. As you can see that although all cells of our body has identical genomes, the transcription status of every cell type are regulated by various epigenetic mechanisms that can occur at pre-transcription level, transcription level, post-transcription level, translation level, and then post-translation level. Thus, all these factors, mechanisms will ensure that each and every cell transcribes a cell type specific genes. Any perturbations in any of these methods leads to dysregulation of expression, which actually reminiscent of a diseased state in general. Okay, I think with this I will end. I completed my talk. If there are any questions that you have, then maybe I'm more than happy to have you. These slides are now I shared with Dr. So you can have these slides with you. I hope it is clear, but if there is something is not clear, maybe you can have these slides with you, and then maybe if you want more clarification.
Thank you very much, Professor Srinivas. I think the session will open it for discussion now.
Okay. Thank you very much.
I request all NRC KRPs to stop the meeting. So if anyone have any doubts, till ask now, the session is open for discussion.
This is very elaborate lecture. I think many new concepts. These are very, very important new concepts. Being teachers, at least I feel that you should really aware of these new developments, so that you know the whole world is now doing research on these kind of areas. So science has gone to beyond the textbook now. So, but these are the reality of life. So I think having this knowledge is very, very important for.
Yeah, it's something very new to us. It's not above our heads, to be frank. That all the pictorial representations in your PPTs is very good, colorful.
Okay.
If there are no doubts from any NRC, I request Dr. M. Madhusupari Madam to propose vote of thanks to Professor Srinivas.
Okay. Thank you so much. Huh?
Good morning, everyone. On behalf of NRC, TVKN, Chittur, I would like to extend our heartfelt gratitude to Dr. Srinivasalu from the Department of Animal Biology, Studies of Life Sciences, University of Hyderabad, for his enlightening talk on eukaryotic genome organization, structure and function. Dr. Srinivasalu's expertise and passion for the subject were evident throughout his presentation, captivating our minds and shedding light on intricate details of eukaryotic genome organization. His insights have not only broadened our understanding, but have also inspired us to delve deeper into the complexities of molecular biology. We are truly privileged to have had the opportunity to learn from Dr. Srinivasalu's wealth of knowledge and experience. His dedication to research and education serves as a beacon of inspiration for aspiring scientists and scholars alike. Once again, we express our sincere appreciations to Dr. Srinivasalu sir for gracing us with his presence and sharing his invaluable insights. We look forward to future collaborations and continued contribution to the field of animal welfare. And also, I'm very happy to know that you are one of my senior at Regional College of Education, Mysore. Thank you so much, sir. And really very delighted and happy to see you on this screen.
Yeah. Thank you so much. Thank you very much.
Srinivas, she's your immediate junior.
Oh, yes, yes. Yes. Thank you.
Thank you very
So
much.
I will leave. Thank you.
I'll be sharing your identity and PPT to
Sure,
all
sure.
the
Yeah.
participants.
Sure, no problem. Thank you very much.
Thank you very much. So, small announcement to all the NRCs. So, today, fourth session will start by 3:45 and it goes to up to 4:45. So last session will be from 3:45 to 4:45 and from 4:45 to 5:15. I request all the NRC coordinators to organize valedictory with the NRC principals. So this is a kind note to all NRCs, please. Thank you very much. So now I request all the KRPs to engage the session online. We'll be joining by 1:50 p.m. for the next online session. Thank you, one and all.
Share this.
Sir, good afternoon, sir.
Sujana madam, your voice is not clear, madam.
Good afternoon, madam. Is it not audible? Is it not audible?
Audible, madam, but it's not clear, madam. Some disturbance is there.
Maybe connectivity defect. I don't know what to do.
Yes, ma'am.
Now, NRC DKW introduce, sir.
Good afternoon. Are my slides visible to you?
Yes, sir. Yes, sir.
Okay, okay. We will join at 2 o'clock. Okay.
Okay, sir. Right, sir. Madam, baby, madam. Now is it all right?
Ma'am.
Is it all right now?
Okay, madam.
Okay.
Okay.
Somewhat okay. Now. Yes. Agent sir, agent sir, list పెట్టాలి. లిస్ట్ పెట్టాలి. అసైన్మెంట్స్ ఎవరు సబ్మిట్ చేయలేదు ఒకసారి అనౌన్స్ చేస్తాం. లిస్ట్ ఎలా చెప్పలేదు. Sir, can we wait for some time, sir?
I have no problem. Whenever you want me to start, I am ready. I am sitting here.
Yeah. Okay, thank you. So I request all the KRPs of NRCs to alert the participants to join the session. Our resource person is already joined us. Sir, police sir, okay, sir. Ramon sir. Good, better. Rajshekhar, NRC DKW. Sir, Rajshekhar sir, am I audible? NRC DKW, Rajshekhar sir. Anyone from DKW? Sir, I just would like to introduce you to the participants, sir. Few seconds, sir. That's actually NRC DKW will introduce you, sir. Introduce you to the participants, please, sir.
డాన్స్ ఉన్నాయి
Manjunath, sir. NRC DKW, kindly respond. Manjunath, sir. Rajshekhar, sir, kindly respond.
Enrichment culture, career description, the magazine of shit.
NRC DKW. NRC DKW.
Madam, Sujana madam?
Yeah, sir, please, sir has joined, sir.
Yes, sir. Sorry, madam. Sorry, madam.
Yeah, please.
I beg your attention to listen the profile of Professor T.H. Venkatramana Garu, who is our speaker of this session. Professor T.H. Venkatramana is a senior faculty member of School of Life Sciences, University of Hyderabad. His main area of research is discovering new bacterial strains, and his theme of research is quest for discovery of new bacteria with novel functions. So far, he published 282 research papers in various reputed national and international journals. Currently, 8 scholars have been working under his able guidance. To his credit, he has 6 patents and 1.76 crore funding for establishing OBD Center for Microbial Informatics. With this brief introduction, I would like to invite the speaker of this session, Professor T.H. Venkatramana Garu. Thank you.
Thank you very much, all the participants over here. It's really unfortunate we are not in contact eye to eye. But nevertheless, I think I will do my best through this online class. Last time we had a wonderful eye to eye contact with all the other participants. Am I audible to all of you?
Yes, sir.
Okay, then it is fine. And I think some of the speakers earlier, like participants, they recommended this title of strategies to cultivate microorganisms. I don't know how many among you are working with microorganisms. Are there any participants who are working with microorganisms? I see hardly any hands which have been raised.
Yes, sir.
Okay. At least one, I'm happy that you are working with microorganisms. You're teaching microbiology?
Yes, sir. Thank you, thank you.
Okay, it's good. It only says that though the others have not raised their hands, it only says that unknowingly others are also working with microorganisms. The one who has raised the hand is professionally trained, probably working with microorganisms. The others are also working with microorganisms, but probably not knowing that they are working with microorganisms. So what we simply say is, for some it is want, and for some it is not want. So we have two categories of people who are always looking for the discovery and cultivating the microorganisms. The other group are those who do not want microorganisms, for example, who are working with cell cultures or animal cell cultures or plant cell cultures, or to that extent, any other aspect. They don't want microorganisms in their what we call cultures. So we simply say that knowingly or unknowingly, each one of us, we are working with microorganisms. It is an accepted fact. I will try to explain to you in coming few slides the significance of cultivation of microorganisms, why we need to cultivate microorganisms, and the various strategies which we are using presently and upgraded strategies which help in the cultivation of microorganisms. I know that most of you, or if not all of you, are teachers who are teaching biology to the undergraduate students. If postgraduate also, it is wonderful. But each one of you can help in the cultivation of these microorganisms, which are the hidden treasure and the wealth of biological systems. The moment we speak about wealth, we only think about the wealth which in terms of money or in terms of gold and precious items. I know that most of you know this and recognize this image, which is from the popular Padmanabha Swami temple, which is considered to be a hidden treasure of our country in terms of wealth. And this is one treasure which says that the god is, or the temple is very rich. But one thing which we also should try to understand, that wealth is not just in terms of gold or precious ornaments or in terms of money. Today we say that health is wealth, and also say that the wealth comes not from these precious gold or ornaments or money, it comes from the microorganisms. Your microorganisms are your health, and your microorganisms are the hidden treasure of your health and the secret of your health. So inventory of treasure in the sixth chamber suggests the value of its gold, diamonds, and other precious metals at cost of rupees 1 lakh crores. But did we ever estimate the treasure what we have in terms of the microorganisms? The microbial treasure is much more important than the treasure what we have seen till now. If I show you one of the slides which speaks about the richness of India in terms of the bacterial diversity, I divide this treasure into the assets and liabilities. Assets are those which we have cultivated and have them as cultures in our laboratory. And the liabilities are those which we presume that they exist in the environment, but we have not cultivated these organisms so far. And if you look at these two, assets and liabilities, we simply see that only 1% or less than 1% of the treasure of microbial wealth is cultivated. Roughly around 99% of the treasure of the microbial wealth is not yet cultivated. This wealth needs to be properly inventorized and modulated for the next generation and for the better tomorrow. So let us try to understand which are those techniques which we basically use in cultivating the microorganisms. So basic techniques required to study microorganisms is to grow or cultivate microorganisms, isolate microorganisms, grow microorganisms in pure culture, observe microorganisms, and the fifth one is to identify these microorganisms. Each of these studies are critical and very important to understand the wealth of the microorganisms. We normally use different medias for cultivating the microorganisms. These are some of the examples which I have given here are the most popular and most common media types which are used for the routine cultivation of the microorganisms. Media such as tryptic soya broth, tryptic soya agar, these are called as general purpose media because they support the growth of many microorganisms. We have very specialized media, for example, blood and other special nutrients, which adds to the general purpose media. This encourages the growth of fastidious heterotrophs. These special or tight media are called as enriched media. Good example is your blood agar. Selective medias, it favors the participation of a particular organism. For example, addition of bile salts or dyes like basic fuchsin or crystal violet favors the growth of gram-negative bacteria by inhibiting the gram-positive bacteria. The examples of these are nothing but your McConkey agar, which is helpful in the cultivation of Escherichia coli. We have differential media, which help in the distinguishing between different groups of microorganisms and even permit tentative identification. For example, McConkey agar or EMB agar, eucine methylene blue agar. These are some of the traditional and classical medias which are commonly used. Even for those who are teaching the undergraduate microbiologists, you try to use some of the medias for cultivating the various microorganisms. If you take the microorganisms and see under the microscope and count the number of microorganisms under the microscope, and simultaneously you serial dilute the sample and plate them on an agar plate and try to count the number of these microorganisms under both microscopic and the cultivated on the plate. These two will never match each other. The count under the microscope is much higher than the count on the Petri plate. The disturbancy or the anomaly of these two are called as the plate count anomaly. While on the Petri plate, you will see that the microorganisms which are viable and cultivable on the plate are the ones which are proliferating, while what you see under the microscope represent the sum amount of all the cells, which includes the viable, non-viable, dead, and also those which cannot be cultivated on the Petri plate. So we call this as the great plate count anomaly. This anomaly is the basis to understand the rich diversity of microorganisms which you come across under the microscope, which only says that on Petri plate, we are hardly able to cultivate the vast majority of the microorganisms which we are seeing under the microscope. There are a large number of factors which are influencing the cultivability of the microorganisms. Identification of substrates and growth conditions which are required and favor the growth of the organisms. Restoration of dormancy. You will see that majority of the cells are dormant in natural habitats. Only under the favorable conditions, these organisms will grow under the laboratory conditions. There are microorganisms, even with the highest nutrients what you provide, these require a co-partner, which we call it as symbiotic interdependency. That means they can be cultivated only when there is another co-partner. Physical contact and spatial proximity is one more factor which influences the cultivability. Physicochemical environmental conditions, pH, temperature, and several other factors are required and influence the cultivability. Low abundance and competition also play a very important role. While those who are working with microorganisms and also teaching microbiology, they must have taught the serial dilution and plating. When you serial dilute and plate, you will see that a large population in terms of colonies appear on the Petri plate. And you try to enumerate these organisms based on the colony forming units. But if you try to incubate your plates for a longer time, you will see a marked difference in the numbers, not only in terms of their incubation for longer periods, but you'll also find some of the organisms which are appearing only at the higher dilutions, which are not seen under the lower dilutions. These are slow growing organisms. When you're trying to use the serial dilution and plating, try to see the slow growers and also the fastidious growers at the higher dilutions, and don't just take the lower dilutions for counting the number of colonies. What are the different strategies for cultivating microorganisms? The classical cultivation strategies and methods, which simply uses the serial dilution and plating on the Petri plate. You will pick up the colony and put the colony on the selective nutrient medium and try to separate this based on various conditions like temperature and pH. And also use certain selective inhibitors if you are specifically interested in one group of organisms. A good example is your leucine methylene blue agar. This is the classical strategy which is used for the cultivation of most of the microorganisms. The other one is serially diluting to an extent wherein you will find only one type of colony. We simply call it as dilution to extinction. You also have another method where you try to separate the cells based on density-based separation. The other cultivation strategy uses the separation of the cells based on the size of the cells. You simply take membranes of different pore sizes and pass the culture through that membrane, and you will see that the cells get separated based on the pore size. The larger the cell size, they will be separated easily based on the pore size. You also have motility-based enrichments. Nice examples are spirochetes. What you do here is you use a very soft agar, and the bottom of the soft agar, you put the media which is having some nutrients. On the top of that, you simply put a soft agar and now put the culture on the top. Based on the chemotaxis, all those cells which are attracted towards this are moving towards the bottom of the culture. It is not only the chemotaxis, but it can also be any other taxes, including your attractants as well as repellents also. So these are some of the classical strategies which are used for the cultivation of the microorganisms. Now, what has resulted in these classical strategies of cultivation? The strategies which we have discussed just now has resulted in the cultivation of only 5 major phylum members. The 5 big phyla extensively studied in India. In fact, it is also globally. The cultivation strategies which I have explained you till now, all these cultivation strategies have resulted in the cultivation of only these members which belong to 5 different phylum members. Which are those 5 phylum members? The first phylum is Pseudomonidota. The Pseudomonidota, also called as proteobacteria, probably most of you might be knowing the proteobacteria. The examples of these are Rhizobium, Pseudomonas, and many other common bacteria. The second phylum members, which are easily cultivated, belong to Basileota, or previously called as Firmicutes. This resulted in the cultivation of Bacillus, Streptococcus, Staphylococcus, and some of the common Firmicutes. The third phylum members are those which belong to Actinomyceta, also called as Actinobacteria. Good examples of these is Streptomyces and related members, easily cultivated on the petri plate. The fourth phylum members are the members of Bacteroidata, also called as bacteroidates. A good example is bacteroidates. The final example is cyanobacteria, which can also be easily cultivated with the strategies which I have explained to you. Example of the cyanobacteria are Cyanococcus, Mashcocladus, and many other things, including Anabina and other things. Now, what we refer today, the classical strategies of cultivation, what I have explained you in the previous slides, has resulted only in the cultivation of these 5 phylum members, which today we refer them as petri weeds. These are petri weeds like your Parthenium, Cuscuta, and many other Ecornia and many other weeds. They do not have any specific lifestyle. They are tuned to the needs and the no requirements of any fastidious conditions. As a result, you take soil and put it under any of these cultivation strategies which I have explained to you with the nutrition, whatever I have explained to you, you are bound only to get these 5 phylum members, and most common are your Pseudomonas, Bacillus, Streptomyces. Bacteroidetes is a little difficult. Synechococcus is also a little difficult. But then these are the 5 phylum members you are going to get. And these today we refer as petri weeds. Now, the take-home message is the 5 are not the ultimate. Diversification should be the future goal. Choosing the easy and leaving the difficult will not pay for novel discoveries. The one what we are referring, that only 1% of the wealth is under cultivation. 99% of the wealth is yet to be cultivated. Using any of these strategies, we will finally land up only in cultivating the so-called petri weeds, which account to the 1% of the cultivated members today. So what are we supposed to do? Schematic representation of the 4 main categories of cultivation strategies. This is a prokaryotic black box, which speaks about only a small percentage of the organisms which are under cultivation. A large number are yet to be cultivated. So people are using different strategies which are modified from the present protocols. I will not go into the details of these protocols, but simply understand that many new strategies have come up in the cultivation of the microorganisms, which includes high throughput cultivation, cell-cell interactions, refinement in the media and nutritions, and in situ cultivation. High throughput cultivation is something you are cultivating in a small chip-like structure, which accounts for about 100 to 1,000 wells. And in a short area, you are covering highest number of medias, whereby we will be able to use different media and nutritional conditions which can promote the cultivation of some of the yet to be cultivated organisms. In the cell-to-cell interactions, we say that large number of these microorganisms are always associated with other organisms. If the co-partner does not exist, they will not survive. The co-partner can be a living one, or it can be also some of the signaling compounds, chemical compounds. Refinement is modifying your media. This is media which simulates the natural medias. For example, the gut. The gut provides a media which is different from what we are providing. So in the refinements, we try to understand what are the refinements which we can do for the media and formulate new medias. So diversification of media formulation, what we are going to speak in this. In situ cultivation, what you're going to do here is try to cultivate the microorganisms under in situ conditions. If I want to grow an X microorganism, which cannot grow without the other organism or environment, I will be cultivating that microorganism only under in situ condition. These are a few strategies which I have mentioned are different from the classical and traditionally used cultivation strategies. There are many more strategies. Let us try to see the reasons for unculturability. Many a times, knowingly or unknowingly, we throw nutrients in the media and ask the microorganisms to grow without understanding the media requirements of the microorganisms. Too high nutrient content, especially organic carbon. Many of the microorganisms are organophobic. They do not like organic carbon at all. They do not like, I will give you example, glucose. Many microorganisms do not like glucose, which we think that glucose is the best substrate for the cultivation of microorganisms. It gives immediate energy. There are large number of microorganisms which are glucophobic, and they are glucoallergic also. So we say that organic compounds play a vital role in modulating the cultivation of your microorganisms. Many may be living in consortia, dependent on each other for their growth. They would like to have a co-partner for their growth. We have a large number of microorganisms which are happily surviving in the presence of the host, but once they come out of the host, they enter into a dormancy state. Those cells which enter into the dormancy state, we call them as viable but not cultivable state, or simply VBNC. I give a simple example of Vibrio cholera. Vibrio cholera is a good example where the Vibrio cholera is hyperactive in the host, but once it comes off the host, within 3 hours, it goes into a state of dormancy. The reasons for this are many, but one simple thing is that all these cells are physiologically and metabolically active, but cannot or do not proliferate and grow in the media what we are providing. Bacteria that otherwise form colonies may present in a dormant state. VBNC, upon exposure to favorable conditions, can again be cultivated. Small invisible colonies and low turbidity obtained, not visible to naked eye. Many a times when we see the Petri plate, we are focusing only on the big colonies, what is visible to the naked eye. Have you ever thought what would be the minimum cell number which can be easily seen by a naked eye? The number of cells per colony, roughly around 10 to the power of 5 to 10 to the power of 6 colony cells per colony are the ones which are visible to the naked eye. But there are colonies on the Petri plate which are having cells which are less than 10 to the power of 5. And these colonies are not visible to the naked eye. These colonies are not as big as those colonies which are greater than cell numbers which are greater than 10 to the power of 8 and 10 to the power of 9, and that big colonies which we are focusing upon. There is wealth in front of you, but we are ignorant of the wealth because we are fooled by the big colonies, leaving the tiny colonies. And majority of the time we are focusing only on those big colonies, leaving the tiny ones. These tiny colonies, which are not visible to the naked eye, we call them as cryptic colonies. These cryptic colonies are much more important than the visible colonies which we see with the naked eye. Mind you, next time when you're all observing the colonies on the Petri plate, try to concentrate a little bit more on the Petri plate to observe these tiny colonies, which most of the time we ignore these colonies and focus only on those big colonies. There are several new techniques for cultivation. I will just try to give one or two strategies for cultivation. You take the sample from terrestrial, freshwater, marine, or host associated, and you enrich the sample. The enriched sample, you extract the DNA and analyze the whole DNA, what we call it as meta-DNA or metagenomic DNA. The metagenomic DNA gives the holistic information about all those organisms which have been growing in the medium what you have provided. In the second step, you will try to use different medias and put this enriched sample in different medias. In some medias, some organisms grow, and some medias, some organisms do not grow. Selectively isolate organisms with specific functional characters. You can cultivate these in different medias with different. Media need not be only nutrients. Medium can also be abiotic factors like temperature, the pH, alkalinity, salinity, blah also. This can help to some extent to cultivate some of the yet to be cultivated organisms. We also have cell sorting based cultivation, wherein you try to use such specific probes for specific group of organisms, which is tagged with a fluorescent DNA. The DNA is not fluorescence. We have a DNA which is tagged to a fluorescent dye. And that dye specifically binds to a specific group of organisms that can be used for separating the cells. We can also use membrane diffusion based cultivation, which also includes iChips. iChips is the name sounds very funny, but very simple. All of you can do this experiment in your laboratory. This is nothing but a tip, micro tip. You just take a micro tip. To that micro tip, you add a few glass balls, small size glass balls. On the top of the glass balls, you put soft agar with a specific nutrient. And this organisms which have this chemotaxis rushes to this microtip and goes into the soft agar, and that you can take that soft agar and use it for cultivation. Anyway, there are a large number of strategies which can be used for cultivating the yet to be cultivated. I have just shown you this slide, how a Petri dish, the great Petri invented this Petri dish, how it has evolved from the past to the present. This discovery of Petri plate goes way back to 1880s. And subsequently, people have changed and evolved with different strategies like micropetries, microfluidics. And today we have this micropetri chip. You all know what is the size of the Petri plate. It is a 4 inches Petri plate. The diameter of the Petri plate is 4 inches in diameter. This was designed based on the needs, not just simply designed based on the, not based on the needs. Now we have these micropetri chips, which are micrometers in size, roughly around 4 to 5 micrometers in size. See the size reduction of the Petri plate today. Hundreds of these micro Petri chips are embedded in a small block. Each Petri chip is a replica of your Petri plate. We have reduced the size of a 4 inches plate to a 4 millimeter plate, which is a revolution in the Petri plates. This 4 millimeter Petri plate can accommodate hundreds of such plates in a small chip. Each of these Petri plate or Petri chip is embedded with different medias. And on the top of this, what you see here, these are the organisms which are put on the top. Based on the need of the nutrient, they migrate to that particular Petri plate. From the bottom, you can remove all these Petri plates and now try to do your analysis. So from a 4 inches plate, we have moved to a 4 millimeter plate, which is far, far superior than what we are using in the laboratory today. This is an important strategy to cultivate the microorganisms. Let me just tell you about the dormancy. Dormancy of microorganisms is very common. And as a result, most of these become non-cultivable in the laboratory. The synonyms of VBNC include dormant, quiescent, ultramicrobial, mini cell, cryptically growing, slow growing, resting cells, or sleeping cells, and one more important terminology, that the cells have gone into a state of coma. We are speaking about coma only for human beings and animals. Do you all know that microorganisms also go into coma state? The coma state can be reversed or cannot be reversed. How long it takes to reverse is a big challenge. Like doctors, how they work on these coma people, we microbiologists are also trying to understand what makes these microorganisms to go into a state of coma and what triggers them back to revert from coma. How long they will be in the state of coma and how long they will take to come out of the coma. Oh, this is really a challenging one. I've given you a wonderful example of Vibrio cholera, which most of the times it speaks about the dormancy. Okay, you have adapted some strategies to cultivate the microorganism. The next is to identify the microorganisms. What best strategies we have to identify the microorganisms? These are the people who are always involved in the identification of microorganisms. The first group of people are the systematics people who will not look into the culture, nor they will look into the organism. They will only speak about the DNA or the most popular DNA gene which is used is your 16S rRNA gene. These systematics, most of the time, based on the sequences, they speak about the identification of the organisms. And each of these sequence is called as operational taxonomic unit or that sequence represent a candidate's taxonomic unit. This is done by most of the systematists. We also have some people who are clinical microbiologists, and the clinical microbiologists always concentrate the clinical identification of the microorganisms based on certain characters which are exhibited by the microorganisms. The characters are nothing but the phenotypic traits. These are all non-taxonomist based on certain characters, biochemical characters, physiological characters, or morphological characters. They simply identify these microorganisms. Neither the systematist nor the clinical microbiologist identify the microorganisms in a proper way. It is only a very few limited core taxonomists who identify the taxa at the level of a species or at the level of subspecies, and they are always trying to identify the new members and describe them as species no, or genus no, or family no, or sometimes phylum no. What I mean to say that identification of the prokaryotes is not as simple as that of as those of the identification of your flora and fauna. The major problem associated with the identification of these prokaryotes is a lack of sexual reproduction. This makes the definition of the species of the prokaryotes much more complicated than for the definition what we use for the eukaryotic systems. Pure cultures. The moment you have a colony, you will say that I have a colony which represents a clone of cells and the culture is pure. Now, the immediate question comes to a microbiologist like me that how pure is your pure culture? Although it is possible to identify microorganisms and even individual cells in a mixed culture, pure cultures are usually used for the identification. Identification is never done by or for a mixed cultures. The immediate question is how pure is your pure culture? Selection of a single colony from a plate does not necessarily assure purity. Especially true if selective medias are used. Live but not growing contaminants may often be present in or near a colony and can be subcultured along with the chosen organisms. You are using a particular media where the contaminant may not be growing there. Because it is not growing, we cannot define that as a pure culture. Friends, it is very important to have a pure culture. And to have a pure culture and to maintain a pure culture, it is not that simple. So what are the quality checks we do for the purity of the culture? There is nothing like I isolated a colony and that colony is pure for sure. How pure is your pure culture? This question always remains. And once a pure culture you have obtained, there is no guarantee and warranty that it will be pure forever. Bacterial quality check is the most important aspect in biotechnological research and often neglected aspect. The quality check means monitoring the purity of the bacteria which is being used, establishing the identity of cultures. These are the two prima facie for a microbiologist to work with pure cultures. Quality checks should be made not only at the beginning of the research work, but during every stage of research I know that most of you have done your PhD, if not all of you have done your PhD, but this is a very important aspect for those who are working particularly with microorganisms. Monitoring the purity of culture before and after experiment is very important. Bacterial quality check ensures the quality and consistency of the results. Maintaining a pure culture stock in reserve is the most important one. Most of us, what we do, once we have a culture, take the culture, transfer it, give it to the other friends, give it, other friend again takes it. Oh, it goes on. The best thing is preserve the culture and use fresh culture every time instead of using a culture which is being handled by many people or an individual many times. This is very, very important take home message. So we say that this is a sign which is used for purity, and pure for sure is assured. But in respect to the microorganisms, there is no purity for sure. One has to be very careful at every step of your experiment. The single colony selection of a colony plate does not ensure purity. Non-selective media should be used for checking purity. If selective media are used, live but not growing contaminants may often be present in or near a colony. Even the non-selective media, apparently well-isolated colony, should not be subcultured too soon. Some contaminants may be slow growing and may appear on the plate only on longer incubations. With bacteria that form extracellular slime or that grows as a network of chains or filament, contamination or contaminants often become firmly embedded or entrapped and are difficult to separate. Example, for those who are working with microalgae and cyanobacteria, this is much, much more difficult. And what you are working, even with the plant systems also, remember that no plant system or no animal system is independent of microorganisms. They are all associated with microorganisms. It is a whole system you're working. It is not the plant alone, plant along with the microorganisms or microbiome. Animal along with its microbiome is the functional aspect. Evidence of purity on solid media, all the colonies are similar to one another, except you have exception. I have given an example of soft and rough variants, capsular variants, pigmented or non-pigmented variants, which may be selected by certain media. Temperature and other growth conditions can also help you to separate and see how pure is your pure culture. Morphological observations can also help you directly observing them under the microscope. You may be able to say that this is a culture which is almost pure, but neither of these things will help you in identifying the purity. So to what extent one wants to identify? Clinical identification. Taxospecies are identified based on morphological, biochemical, physiological, and serological characters. Polyphasic identification is done by the taxonomist. As a result of clinical identification, we have large number of pseudo-identification of pseudo-taxonomists. You go to any pathological laboratory and give your urine sample or blood sample, they will simply stick it on a media and say that your urine, you have Escherichia coli infection. You have, for example, you give your what we call the fecal material. Immediately they say, well, that it is you are having infection of Enterobacter. Likewise, clinical identification where these clinical microbiologists do it. Neither of these are done in a proper way to identify the right taxonomic status of these microorganisms. We call them as pseudo-identification or pseudo-taxonomous. So what we do for the identification of the microorganisms is very simple. Once you have the colony, the first step what you do is do the Gram reaction. Gram reaction is the most simplest technique even for an undergraduate or a postgraduate class to teach what is Gram positive and what is Gram negative. The other identification what we do is we ask the students to see under the microscope and record it as rods, cocoid, spherical, or whatever morphology they have. These are the two classical protocols for identification of the microorganisms, particularly the bacterial systems. So bacteria are slightly negative, so are attracted by the positive chromophores of the basic dyes. Which are those common basic dyes we use? Crystal violet, methylene blue, safranin, and basic fusion. And we follow a simple protocol which helps in differentiating the bacteria as gram stain positive or gram stain negative. And some organisms like your Mycobacterium, which are recalcitrant cell wall, we use acid fast staining and simply say that acid fast positive or acid fast negative. This has resulted in a very simple classification, which is primarily based on the cell wall. So these microorganisms, the prokaryotes, have been segregated as gracilicutes, firmicutes, tenoricutes, and mendolicutes. Most of the textbooks give this as the primary classification of the prokaryotes. Gracilicutes, thin skin; firmicutes, strong skin; tenoricutes, soft or tender skin; mendolicutes, skin with faults. And this is how classification follows. And if you see the hierarchy of this, it is the species, genus, family, class, division, and kingdom. And this whole classification is primarily based on the cutin or the cell wall. But when you see, there are a large number of limitations of gram staining. I will not speak all the limitation, but I will just give you an example of a cyanobacterium, which has roughly around 120 nanometers thickness of the cell wall. But yet it gives gram stain negative. When we are teaching the students, we teach that this crystal violet goes inside. You add a moderant like your iodine, which strongly binds to the cell wall. And subsequently, you add acetone or alcohol, which helps in increasing the pore size. And when you add a counter stain like safranin, that binds to the, again to the cell wall. And during the process of washing, whole of crystal violet goes off. This is so simple and explains probably to a common man, the principle is not as simple as what we are thinking today. This principle is much more complicated than what we have been teaching to the students. Nevertheless, I will just give you cell wall is nothing to do for a gram stain positive or a gram stain negative. We have a large number of organisms which have a very thick peptidoglycan. I have given a wonderful example of a cyanobacterium, which has roughly around 120 nanometers thickness of the cell wall, but yet it does not retain the crystal violet. Now the question is what we are teaching students, gram staining principle apparently appears incorrect. I have just given you some examples of the limitations of the gram staining. Those who are interested can go through these gram staining limitations. A large number of these are the limitations of the gram stainings. With regard to the species concept, I just explained to you that prokaryotes, defining a species of a prokaryote is not as simple as those of the eukaryotic organisms. Bacteria are morphologically identical, but physically they are different. They keep on changing their genetic composition. They can easily acquire the characters of other bacteria by taking their genetic material. They are very flexible at releasing and picking up the genes of other bacteria, unlike your eukaryotic systems. They lack true sexual reproduction, but it occurs through another process known as recombination. Their biochemical functions are totally different, even the individuals belonging to the same genus. These are a few limitations of defining a prokaryotic species. We say Escherichia coli simply, but if I ask you, how do you define a species of a prokaryote? There is no answer. But how do you define a species of an eukaryote? We simply say that these are sexually competent. There is nothing like that in prokaryotes, which makes the definition of the prokaryotes much more difficult than what we have today for the eukaryotes. Now, these prokaryotes are defined based not on their morphological features, define these members as species based on certain genes, particularly the rRNA gene, and we define those isolates that have greater than 97% identity of 16S rRNA gene with a good length of your 16 rRNA gene. These might be represented as the same species. Mind you, it is also the DNA-DNA hybridization of these two organisms should be greater than 70%. And those organisms which have less than 97 or 98.7 16S rRNA gene identity and also has less than 70% of DDH, we call them as these are distinct species. Species which are having 16S greater than 90% identity, but having DDH less than 70% identity, they are also defined as different species. And we call them as genomospecies. Genomospecies are those species, I repeat, are species which are having 16S rRNA gene identity greater than 97 or 98.7%, but have less than 70% of your DDH. These are defined as genomospecies. The 16S identity does not delineate both the members, but the genome, the species is separated based on their genomes. We call them as your genomospecies. So based on this, the phylogenetic classification has revamped the simple gram stain classification, and today's hierarchy speaks about the domain bacteria. We don't have the kingdom, the phylum, class, order, family, genus, species. And those which are less than the species, we call them as microdiversity, which represents as subspecies and your varieties. Most of you have heard about seroverse, pathoverse, xanthobacter Oryzae, Pathover oryzae. This is not a subspecies, it is a microdiversity which has adapted to its pathological character, serological character, serovers, biological or biochemical differences, biovers, phage differences, phagovers, and so on and so forth. These are all the organisms, species less than the species. So the hierarchy now is based on phylogenetic affiliation. So today's identification of the prokaryotes is not simply based on the gracilicutes, what we have defined based on the cell wall, but it is based on multiple approaches, which includes the expressed characters, genetic characters, and the phylogenetic affiliation. Simply in a nutshell, we call it as polyphasic taxonomy. The prokaryotic taxonomy, including the taxonomy of the fungi and taxonomy of the higher things, higher plants or animals, is also now slowly approaching to the polyphasic taxonomy, not simply biological characters alone. So how can maintain the purity of the cultures? How good is your laminar flow chamber and workplace? All of you, particularly those who are teaching, you will say, go to the principal and ask the principal to give a laminar flow. I tell you, laminar flows are the most dirtiest places for cultivating the microorganisms. The laminar flows gives overconfidence of the student to work in a laminar flow. Laminar flows are the most contaminated places in your whole lab. Have you ever checked the quality of the air and maintenance of aseptic conditions in the laminar flow chamber? Aseptic handling skills are essential. Let your students work only in front of 2 or 3 flames. Once they are able to handle the organisms in front of a flame, I am telling you, they can easily handle the organisms even in a laminar flow. Just having a flame in front, what we call it as Agni Sakshi, and inoculating will not ensure aseptic handling. Your hand and the air you exhale are excellent sources of microorganisms. You will never be able to handle a pure culture despite all the best facilities you give, unless you're handling the organisms. That is nothing but your art of handling and cultivating the microorganisms. One has to learn the art of cultivating the microorganisms to become a good microbiologist. So I will slowly end my talk by explaining how best you can preserve your cultures. Very simple. You periodically transfer and refrigerate at 4 degrees. Let me also caution, most of these, at least some of these microorganisms, do not like cold temperatures. As a result, they go into a dormant state, which we already defined as VBNC state. You should be knowing whether your organism can be stored at low temperatures or not. Even 4 degrees is sometimes toxic to the organism. Mineral oil slants, which we commonly use for fungal cultures, can also be used for microorganisms. Sometimes glycerol is also used for preserving. Freezing in growth media with glycerol, which protects from osmotic shock. Lyophilization for long-term preservation. Ultra-cold freezing with liquid nitrogen. These are some of the simple ones. But very important is periodic transfer and refrigeration. Very, very simple protocol. Anybody can adapt in a common laboratory for preserving the microorganisms. All of you are the future trainers for microbiologists. As I told you in the beginning, for some it is want and for some it is do not want. Irrespective of want or unwant, you are bound to work with microorganisms. Precisely, you should be disseminating this knowledge to all the young minds. Most of the times we go for sample collection and we try to teach the students the importance of cultivating the microorganisms and also protecting the microorganisms. Finally, I would like all of you to take home the message. The microbial wealth is not yet discovered to its potential. We need to protect our microorganisms, like how we have recorded the Red Book, endangered species of plants and animals. We really do not have any records of the past, present, and forget about the future. Once a microorganism leaves, you cannot regenerate that microorganism, and the rate at which we are losing the microbial wealth today is amazingly very fast. Like how we are protecting the macro life forms, there is an immediate need to protect the micro life forms. And this is only a small lecture which I thought that all of you will take this message to all your students to tell and speak about the importance of microbial wealth. One small microorganism like corona has made alert to all the life, all the human life, where even a layman today speaks about corona. And also he is aware of microorganisms today. The good thing what corona has done has taught microbiology to one and all. Thank you, all of you, for your patient hearing.
Good evening, sir. Thank you for enlightening us with the latest pure culture isolation techniques like microplate chips, microfluidics, and micro techniques. So, how does this microplates work, sir? Can you give us a basic video and differential packet time to grow the microorganisms?
Microchips are ready-made available. You and I cannot prepare these microchips. Even the Petri plates are not prepared by us. They are manufactured by some of the people which are buying and using it. Even the microchips are manufactured by some of the manufacturers.
Yeah, what is my doubt is, are there already fed with the microorganisms, basic video and differential videos? Each chip has its only one type of microorganism.
See, you will get the chips. You can buy the chip. And in this chip, in each of the well of this chip, you will be adding the of your choice.
Yeah.
So that will promote the growth of specific microorganism.
Yes, sir. Okay, sir. Thank you, sir.
These chips are available, but not along with the media. You had to tailor your media and put that media into the wells.
Okay, okay. Thank you, sir. Any more doubts from any participants? Sir, we have already shared your mail ID, sir. They may contact you.
No problem, madam. You're always welcome. All of you are always welcome.
So, if no more clarifications, I request NRC DKW, Rajshekar sir and Manju sir to propose vote of thanks to Venkatraman sir. NRC DKW.
Don't worry about vote of thanks, madam.
No, sir,
Let
no,
me
sir.
thank all of you. Let me thank all of you, and
Because
wish that
No.
some of you are definitely benefited by this lecture
No,
and
sir.
pass on the message to the others.
No, actually, they have some connectivity problems, sir. Now I request Rajashekar sir to propose.
Madam, hello, madam. I'm here only, ma'am. Madam, shall I proceed, ma'am? So, I mean, I'm very much privileged to have been given this wonderful opportunity to propose. Thanks from all the corners of this 5-day TOT program. The eminent really, it's a wonderful session. So by all your, message, we have been enlightened from last, 1 hour or so. So it's really enlightened, sir, all of our participants. And you touched all the corners of your research. It's really very interesting, sir. With all the efforts, by taking out your valuable time, you spent, meaningful time with all our participants, and you also clarified their doubts. So on behalf of all the 9 NRCs, it is my privilege to convey my deepest thanks to you for your great and very enlightened lecture, sir. Thank you. Thank you very much, sir.
Thank you, all of you. Bye. All the best.
Thank you, sir.
Thank you.
So we will be having another online session by 3:45 p.m. now. So till then, I request KRPs to engage in offline giving activities. So as we talked yesterday regarding LMS, we'll be sharing a Google form. Kindly share it to all the participants, please. Please. 340. Welcome back to the next session. I request all the NRC participants to join immediately. Our next resource person, Swami sir, has joined already. I request all the NRC coordinators and the KRF to inform the participants to join the session immediately, please. To once again request all the NRC coordinators and KRPs to inform the participants to join the session immediately, please. Our resource person is ready for the presentation now. NRC PR College. Sir, Dr. Srinivas sir. Sir, Dr. Srinivas Logan. NRC Kakinada, kindly respond.
Yes, ma'am. Yes, ma'am. We are responding.
Yes, sir. Are you ready, sir?
Okay, ma'am. Okay, sorry. Come on.
Sir, I think so most of the participants joined. So I request you to invite the resource person by introducing to our participants, please.
So, good evening, dear participants from all the NRCs spread over the Andhra Pradesh. I deem it as an honor to introduce Dr. J. Swami Garu, Scientist C, Botanical Survey of India, to the galaxy of botanists and microbiologists attending the TOT program from 13 NRCs spread over the Andhra Pradesh. Dr. Swami Garu is holding Scientist C position in Botanical Survey of India. He is working with the Botanical Survey of India for the past 15 years. And he worked in different areas. Particularly, he served in the Deccan Circle Botanical Survey of India at Hyderabad. And recently he joined the Botanical Survey of India, Kolkata headquarters. And he is the man behind establishing the Nature Guide in 2023 and Wild Table Group section in 2024 at agency boards Indian Botanic Garden, Howrah. And he is recognized for his contributions to the hibiscus genus, and he is a member of International Hibiscus Society and published many national and international research articles. And he is presently focusing on creation of a taxonomic garden at Howrah Botanical Survey of India, according to maritime and local classification. And he worked on the sacred groves of Deccan Circle and ecology and the conservation of many species. Presently, he is working on ex situ conservation, propagation techniques, and the taxonomic division of the genus Convergia belonging to Acanthaceae. He is working on the development and categorization of hibiscus cultivars at the Botanical Survey of India, Howrah, and also working on the legumes development at the agency boards Botanic Garden. and development and maintenance of the aquatic plant section at the AGC Coast Center for Botanical Center of India. With this brief introduction, I request Dr. Swami Garu to deliver his talk. So we are very grateful to you, sir, for giving your valuable time and addressing. Thank you, sir. Thank you.
Am I audible, sir?
Sir, you are audible.
Yes. A direct live share presentation. Is it visible, sir, screen?
Sir, it is visible, sir.
Okay. So good afternoon, respected teachers and organizers of the Andhra Pradesh Commissionerate of Collegiate Education. So my name is Dr. Swami. I'm working as a scientist in Indian Botanical Survey of India, Indian Botany Garden. So already explained about my introduction about me. Almost I have been worked almost 15 years in various botanical survey of India regional centers in like Coimbatore, Hyderabad, and now in Kolkata. So almost completed 17 projects. In that, out of the 17 projects, I published more than 130 articles, research papers, including 10 new species, and I wrote 8 books on this, especially in the plant taxonomy and conservation and medicinal botany. So presently, I'm working on 6-7 projects. So some of the students also working under me and this reading program especially. So with this introduction, I will start myself. Today, I'm going to talk about the herbarium technique with special reference to the virtual herbarium. So may I know, sir, how much time I have actually, please?
One hour, sir.
1 hour. Oh, thank
Yes.
you. Okay. So now I am going to cover actually some background of the herbarium and herbarium history and development. So index herbarium and important herbarium in the world and India in our state and type of herbariums and herbarium methodology, role of the herbarium in plant taxonomy and conservation and virtual herbarium. So we know about the herbarium actually. So herbarium is a storehouse of dried and pressed plant specimens collected from different localities and which are mounted on an appropriate sheet. So those sheets are arranged according to some approved system of classification. Here, approved system of classification means there are the different kind of system of classification: artificial system of classification and natural system of classification and phylogenetic system of classification. Any system of classification we can follow for arrangement of the herbarium. Generally, in our India, we used to follow Bentham and Hooker system of classification. Recently, many small recently established herbarium, it's in the recent established herbarium, they are following now APG4 system of classification, because now this is the recent phylogenetic classification system is acceptable worldwide also. So, but like our board herbarium, it's very difficult to follow the APG4 system of classification because that is not in the constant mode, that every year it is changing. So now as for the APG4 system of classification, there are 416 families. Earlier we arranged our all herbarium as per the Bentham and Hooker system of classification, that is 202 families. But now it becomes almost double, 416 families. Again, changing is that this lot of collection and it's very difficult manpower also, the cases of the manpower, we are able to shift those kind of present system of classification APG4. Still we are following the Bentham and Hooker system of classification. So there is no question any kind of system of classification we can follow for arrangement of the herbarium. This is for easily retrieved of the particular specimen in the particular family. For that reason only we have to arrange the particular as per the any system of classification. So actually this herbarium, when it actually was started, this art of herbarium started by the Professor Luca Gini. In the period is the 1492 to 1556. And his work as actually basically is a physician, it's a Bologna University. Then he started working on teaching the students, especially in the medicinal botany. So during the period of 1934, at the time of teaching, he used to collect the plant in the surrounding areas of that particular Bologna University. So after that, he became a professor in the University of Pisa in 1544. And then he attached to the one botanical garden. Then he established this botanical garden. That is the first botanical garden in the world. It was established in 1544 in the Italy. So then after that, he started a collection of evidence specimens. Every day he used to take his students to teach the classes in the field itself. So in the winter time, it's very difficult to take out the students to the field. So due to that reason, he collected the specimen.
Your presentation is not visible.
Not visible.
Your presentation not visible. Share your screen, sir.
Is it moving, sir, slide? The slides are moving?
No, not moving.
Your slides are not visible.
Share your presentation, sir. There is the option to honest.
Again, I will share.
Yeah. It is okay, sir.
Yes, sir. It's visible.
No.
That is F5, sir. You change to F5. Presentation mode.
Srinivas sir, I think it's better we share the slides from your side.
Is it moving well now?
Now they are moving.
Okay, sir. It's okay, sir. Okay, sir. Okay.
Okay. So actually, when he started the working on this plant, he used to teach his students at the garden itself. Then after that, when he attached to the University of Pisa, then in the winter time, it's very difficult to explain in the outside of the field. Due to that reason, he collected the plant specimens and he made the herbarium. First, the herbarium developed by the Professor Luca Gini. He's an Italian professor. Because in the winter time, it's a very difficult Italian condition because of the snowfall. So then he made that herbarium. It's a bundles of bundles. He used to mount like this, 4 to 5 specimens. He used to mount on the herbarium specimen, one sheet of the herbarium. So then they used to call as these specimens the Arta siccus. These bundles of the herbarium they used to call as the Arta siccus. So the first herbarium in the world, so University of Padua, it's having in the 1545. He is developed that herbarium. Then this techniques is later students by Gerard Sibo and Alrovando and Cisalpino. These 3 years started this art of technique, and they are, especially the Alrovande, is almost more than 8,000 natural illustrations he made for especially those kind of plants, whatever are available in that region. So this thing in the mid part of the century, maximum he collected many specimens. Alrovande collected many specimens and they developed in the herbarium in that, especially in the Italy. In the middle part of the 16th century, this technique has started in the England and is spread to the many other parts of the world. So initially the herbarium referred to the book of medicinal plants. Initially the word herbarium is referred to the book of the medicinal plants. Single word is the herbarium, plural is the herbarium. Plural, at the time, when Lucas started this technique, he used to call it the present herbarium, the Arta siccus. The first herbarium word used by the Peter D. Tauniforte in his book Elements in the year 1700. So then after that, earlier they used to make 4 to 5 specimens into the one herbarium sheet. So then anybody want to refer those particular specimen of the particular family, it is very difficult to retrieve that particular specimen. Then after that, what Linnaeus did, he is the father of modern plant taxonomy. What he did, instead of pasting to 4 to 5 specimen into the one plant, one herbarium sheet, we have to arrange one particular specimen to the one herbarium sheet, and we should not go as a group. And we can arrange any approved system of classification. These are the major things contributed by the Carl Linnaeus. Is the father of modern plant taxonomy. So then, if you arrange like one specimen on the one herbarium sheet, then easily you can refer. Then if you bind like a book, very difficult to find out that then taking the specimen, so much damage will be happened. Then if you arrange any system of classification, then easily we can contribution done by the Carlos Linnaeus for this herbarium. Actually, where we find this herbarium? Herbarium actually generally it's associated with the universities and colleges, botanical gardens, museums, and research institutions. So these, where the, especially the botanical work is going on, these particular institution only herbarium is associated. So these all the herbarium, whatever the world, for the past 400 years, they are collected across the globe in the different parts of the country. They are collecting their various specimens. They are making the herbarium and they are depositing their own herbarium. So this particular herbarium, each and every herbarium is having the particular acronym. So these acronym is given by the Index Herbarium. So these Index Herbarium is the global director of the public herbarium. It assigns the each and every herbarium. So like our Botanical Survey of India, if you see our headquarter herbarium, Central National Herbarium, this is the biggest herbarium in India. The acronym of the particular herbarium is the CAL. That is the acronym of the particular herbarium. So this Index Herbarium is maintaining all the world herbarium details in their website. This is the website. It started from since 1996 onwards. It's working. If you click this herbarium, like scienceweb.nypg, near Botanical Garden, actually is running from the near Botanical Garden. So if you enter this link, you will get the all kind of information. This is the link will come. Here you can search. You can based on the, if you know the acronym of the particular herbarium, just you can type the herbarium name. It will come. If you type CAL, if you type the CAL, you will get the all the information where it is available, who is the owner of the particular herbarium, how we have to reach the particular herbarium, what are the specimens they are having in their herbarium, what are the different kind of groups of specimens they are having. So who is the contact person? So all the details you will get it. If you don't know that acronym, just you type the name, like Department of Botany, Herbarium of Andhra University. If you type it, that all that acronym it will come. Then SKU, Sri Krishna Deva University. If you type just SKU, if you type it, it's okay, it will come. Otherwise, if you don't know that acronym of that particular herbarium, if you type complete Sri Krishna Deva University herbarium, the details will come. So with the contact person, then all the details will come. How many specimens there are, every details will be available. The physical location, address, contact number, history, and names and contact information, areas of expertise, who is associated with that particular staff, all the details you will find in this Index Herbarium. So these are the herbarium actually distributed across the globe. Mostly herbarium is distributed in the Europe and followed by the North America. Then very least in the African countries, less herbarium is available in the African countries. So these are the, you can see the world. Among this, Europe is having the highest number of herbaria. So the more than 800 herbaria is having the Europe. So throughout the globe, almost more than 3,324 herbaria is available as of the 2009, according to the editor of the New York Botanic Garden, Barbara. So this is the specimens, more than 392 million specimens across the globe. For the past 400 years, they have collected and deposited in the various continents, in the various parts of our world. So this total is almost 180 countries having more than 3,300 herbaria. It's having more than 392 million specimens. So more than almost 50,000 staff is working with this herbarium. So these are the important herbarium in the world. Coming to the Paris is having the highest number of collections. The acronym of the Paris herbarium is P. This is the P. It's having the more than 10 million specimens. It's located in the France. The Paris herbarium is located in the France. It's more than 10,000 specimens. It's 10 million specimens. It's having. Followed by the Kew Botanical Garden. So like this, so many international herbariums having a number of specimens. Coming to our Indian position is the 30th position. This is the biggest herbarium, our Botanical Survey of India, Central National Herbarium, located in Kolkata. So it's having more than 2.5 million specimens. So this is the biggest number of specimens in the world. Coming to our India, there are 60 major herbariums available in India. So many minor herbariums also is there, but majorly accepted by the New York Botanical Garden Index Herbarium. Major 60 herbariums are there. In this, Central National Herbarium is having the highest number of collection, followed by the Forest Research Institute, Devaradan, then followed by our Botanical Survey of India Herbarium. So coming to the other areas, especially in Andhra Pradesh, so this we have different regional centers across the India. There are almost 12 regional centers. We have 4 regional units. We have almost 16 regional, 4 regional units and 12 regional centers. is in this actually the Deccan area. It's coming to the Odisha and Andhra Pradesh and Telangana. We have one separate regional center, that is the Botanical Survey of India Deccan Regional Center. These Deccan Regional Center cover the jurisdiction of the areas Andhra Pradesh and Odisha and Telangana. Like southern part, if you go to Kerala and Tamil Nadu, that will cover the southern regional center that is available in the Coimbatore. The acronym of the herbarium is the MH, Madras Herbarium. So in the Deccan Regional Center herbarium, the acronym is the herbarium is the Botanical Survey of India Deccan Regional Center herbarium, BSID. So it's having the more than 20,000 specimens in this herbarium. These are the different herbarium, more than 3 million specimens we have in different regions of the Botanical Survey of India. Almost we have digitized in the now virtual mode. It is available. We already uploaded in the website. Many of the specimens is easily access to each and everyone that I will show in the coming slide. So coming to the our state in Andhra Pradesh. So these are the major herbarium, especially in the Sri Krishna Devaraya University herbaria. It was established in 1980. So this is having this is the maximum number of specimens is having this Sri Krishna Devaraya University herbarium. So this followed by the Deccan Regional Center. This is the having more than 25,000 specimens. Then Sri Venkateswara University herbarium. This is located in Tirupati. The acronym of the herbarium is the SUVUTY. Then Usmania University. This is among these all these. This is the oldest herbarium, but it's having a very less collection. This is the actually main taxonomic work was carried out in the 1970 to 80 period. There was the Professor Rajagopal sir. When those worked at that particular area, that time they collected this kind of specimen, including these 8,000 specimens. So many foreign collections also it is there in the Usmania University herbarium. Recently, there is no much work in the from the Usmania University. So very except two to three PhD works. Recently, that was also recently done after 2015 onward. So there are the different kinds of herbaria based on the functions. There's a general herbaria and special herbaria. In the general herbaria, further divided into the international herbaria and national herbaria, local herbaria. In the special herbaria, these are the historical herbaria, herbaria with special research program like environmental impact assessment studies, any environmental related studies, and teaching herbaria, and virtual herbaria, and job related herbaria. These are the different kind of herbaria. One by one we can see. So then the international herbaria, these are actually very large herbaria. Often it's having the more than 4 million specimens. It's having the global representation of this particular herbarium, having the global representation of the specimen. It's a comprehensive, this of the near comprehensive range of taxa as possible. They are very rich in the types, especially in the old collection, old type specimen. They are having a lot of old type specimen. They are having this particular herbaria. So these the major function of the international herbaria is broad scale studies of family and above the level. So family and above the level of the work they used to produce from this international herbaria, and production of the generic monographs and major floras covering to the several countries, especially several countries. So these kind of floras they used to produce by using this international herbaria. So if anybody wants to study those particular herbaria specimens, they can give role and they're providing facilities for visiting as a botanist. We can go and we can write letter to those particular institutions, and we can go, we can study those particular specimen. Otherwise, we can ask them to give on the loan. Sometimes they used to give, but that is based on the NBA guidelines. So if they are allowed, they can give the specimens and we can study and we can send back to those particular area. So these are the international herbaria that see some of the example. Paris herbaria, this is the highest number of collection, more than 10 million specimens. Then followed by the Cube herbarium is having more than 7.5 million specimens. So then near Botanical Garden herbaria, this is having the 6.5 million specimens. So like Komarov Botanical Garden Institute, this is living in Russia, it's having. So this is having more than 5 million specimens. So like this, so many international herbaria it is there. So coming to the national herbaria, these national herbaria generally geographically cover the country and neighboring phytogeographically similar areas. Like if you take the India, Pakistan, Nepal, Bhutan, Myanmar, these are the areas specimen. We have so many plenty of the collection in our botanical, in our central national herbarium. So these kind of national herbarium major function is contributing the floras, major floras covering into the several countries, and production of our national floras. We have actually in 2020 we published the flowering plants of India into the 3 values. So these based on the specimen available in the all various botanical botanical survey of India herbarium, especially in the central national herbarium, these having the highest number of collection. Generally, whatever the collection made from that particular area, so we used to get one set of the specimen we used to receive to the central national herbarium. Now lack of space, we are not taking those particular specimens from the regional centers. Now whatever the specimen is available, that is more than 2.5 million specimen available that we are managing now. So with this particular specimens, we can create the floras at the national levels, and we can produce the divisions and monographs in the particular groups or particular family or genus or families, and we can go for the assessment studies of the endemic and threatened plant species. So whatever the plants you want to assess, if you want, like if you know the IUCN red list criteria, there are the 9 criteria. So as per the 9 criteria, there is extent criteria, extent in the wild, and critically endangered and vulnerable and least threatened and least concern and data. There are the different 9 criteria are there. This is based on the IUCN guidelines. So before, if anybody want to do assess the particular herbarium specimen, before going to the assess the particular species, first we have to consult the herbarium. So what are the specimens collected from those areas we have to document from the herbarium specimens. So then we can take the data, what is the on the herbarium specimen is available data that we have to write. So based on that data we can go to the field and we can verify the plant species, whether that particular species is available in that location or not. So then that kind of data will be helpful for the assessment of the endemic and threatened plant species. So this also includes the same kind of service like international herbarium, what they are having, same kind of facilities is there available. If anybody want to come and visit our Central National Herbaria or any other regional center of Botanical Survey of India herbaria, if you want to write, just you can write one mail, one letter to the concerned head office. It's available in the Botanical Survey of India website. It's accessible to everyone. Just you can write to the mail and either mail or a letter. Then they will allow you can go and consult the herbarium specimens. If you want to work on either floras or divisions or monographs. So this is the Central National Herbaria. This is the building of the Central National Herbaria. So we are arranging, it's like the 5-story building. We are arranging specimens like this. So this is we have also type specimen. We have rich in the type collections. So we have monocot type herbarium and dicot type herbarium. So there are different types of herbariums we are maintaining here. Separate, we have a separate room for this maintaining of this particular type specimens. So second largest herbarium in India is the Dehradun herbarium that is located at the Forest Research Institute, Dehradun. So that is the herbarium, the second largest herbarium. Then after that, followed by the third herbarium, largest herbarium, the Botanical Survey of India Southern Regional Center herbarium. This is located in the Coimbatore. This is the building. So this is the 3-story building. So we arrange in the basic ground floor, we arrange in the polypetale, middle floor gramopetale, and the top floor we arrange in the monocotyledons. So these are the compactors to save the space. Instead of using these almirahs, we use these compactors. So we can so a lot of compactors into we made the 2 stories, and first ground floor and first floor. So top floor normal almirahs only we arranged for monocot herbarium. So easily we can number of specimens we can increase by using these compactors. So another Shillong is the 4th largest herbarium in India. So like this, these are the Indian herbariums we are having. So this is the Deccan Regional Center herbaria. It's located in Hyderabad, especially in the Koti. So in the Koti, it is available. Whoever wants to study, especially on Andhra Pradesh, they can go and they can consult these herbarium. We arrange the specific. This is in the 3rd floor in the Deccan Regional Center herbarium in Hyderabad. So we have almost more than 25,000 specimens available. These major collections from the Odisha and Andhra Pradesh and Telangana. Few collections from the Tamil Nadu and Kerala also is available in this particular herbarium. If anybody wants any collection, because already that particular Andhra Pradesh maximum flora we covered from the when we are in Deccan Regional Center, we covered all the areas of Andhra Pradesh. So all the different parts of the Andhra Pradesh specimen we have collected and we incorporated in the Deccan Regional Center. If anybody wants to consult those specimens, we can go and just write a letter to the concerned head office and you can consult the specimen. So then this is the local. Next herbarium is the local area. Local area is it's like any university, small kind of university and regional small unit. They may have this kind of herbarium. This especially deals with the region within the country, such as a state or district, even much smaller area. Like if you take small national park, small botanical garden, small like zoo park, that those people also whatever the plants available that particular area, they can make herbarium, they can maintain. So these kind of this contains actually there's a very less type specimen. These are especially local herbaria, recently established herbaria. They don't have much herbaria. If whatever they are describing the new species, they used to deposit it in the less national level herbaria like Central National Herbaria in the Kolkata. So all the significant taxa should be represented but not necessary to keep large number of each taxon. These particular areas, what are the regional area of specimens is available that they used to collect and deposit that particular area. The function of the local herbaria especially contributing to the national flora. When we are writing national flora, these kind of herbaria will be helpful for the local distribution of that particular plant. Then production of the local floras like state floras and each district floras and protected areas floras like wildlife sanctuaries, national floras and tiger reserves, like that floras also begin produced by based on using this local herbaria. We can produce any kind of checklist by using this local herbaria. These also having the same kind of services. If anybody want, you can ask if you want a particular, if you revise in particular family, you can ask the specimen, the concerned family specimen. They will give a loan after studying the specimen. You can hand over to the again institution. So this is one of the example of the local herbaria, the Usmania University herbaria. The acronym of the Usmania University herbaria Hyderabad is HY. So these are the specimens of the specimen is described from the olden days, like in 1980 period, Ebenezer Ramayana. It's collected from the Professor Ramachandra Jari and from Maguna district, and he named as to honor Professor Ramaya. They honored as the Ebenezer Ramayana. So Lycocarpa Reddy, this is also collected from the Nalgonda district. So Satyanarayana Reddy are collected from this particular specimen when he is going to the Nalgonda, and the roadside he collected in name as a actually he unable to identify this particular specimen. Then he sent to the foreign in the Q herbarium. In the Q herbarium, he identified the particular species, and then he named it to the collector name Satyanarayana Reddy, and his name he named as the Lycocarpa Reddy. So these are the few of the collections, olden days collections from the Osmania University. So these are the some of the local areas. So then coming to the special herbaria, there are the different types of special herbaria is there. So for example, one like historical herbaria. These historical herbaria, especially in the old, very old collection, heritage collection. These kept actually separate as the herbarium between the general herbarium, or like Polish herbarium in the queue and Decondal herbarium in Geneva. I believe a separate institution outside is there for this particular historical collection, like Linnean Society of Denver. This is the Linnean Society herbarium. It's available in London. These are the Linnean collection. Whatever is Linnean collection is there, those collections they are maintaining as a separate herbarium. Like Decondal herbarium at Geneva, they are maintaining within the herbaria in a separate section, like this historical collection. These are the collections generally they may not allow for the public people, normal public. If any special reference, if you want to see there's any kind of taxonomical problem, with special permission only we can consult this historical herbarium. So coming to the one of India, so one of the oldest specimen is collected from the, especially in between the Andhra Pradesh and Chittoor area and Tirupati, there is Tirupati and Madras area. This is the specimen, one Cypressy family specimen is collected in the 1696, June 1696, collected by the Dr. Samuel Brown, a British surgeon. So this is the collected, this is the oldest specimen. As of now from our India, this is the oldest specimen collected from this Madras and Tirupati between area. So the name, actually this particular species is given by James Pettifer. So earlier you can see the names, how they used to give Cyperus, Ilicinus, Medius, Canicula, Sasa, E. Madras Padma. This indicates that the name is a very so lengthy name. This is actually the polynomial name. So after that polynomial stop, now binomial nomenclature we are using. Now current binomial nomenclature of this particular species is Cyperus crocus. Cyprus crocus. As of now, this is the oldest specimen from our India. It's collected in June 1696. So teaching herbaria, these kind of herbaria, especially in all universities and especially in any college, you can collect this particular specimen. Especially if you want to, when you are going to teach in the taxonomy in the particular family, those kind of specimens always may not available throughout the year. If you collect this particular family, if you take like Malvaceae, what are the species of the Malvaceae available? That if the particular season if you collect and make it herbarium, when we are going to teach to students, that is easily it will be helpful for the to exhibit those specimens to the students. Then easily they can understand the particular specimen because those specimens may not available throughout the year, especially herbaceous. So these kind of teaching herbarium, especially it's available in the colleges or universities. They are maintaining separate herbarium in the teaching herbarium. So then herbarium is for special research program. Whoever want to studies like anatomical studies or cytological studies or chemical studies or ecological surveys or environment impact assessment studies or host insect plant animal interactions and host plants insect pests, then any microbial studies, So these kind of studies, who are performing these particular studies, they used to collect the specimen. But these specimens are not welcome to deposit the particular general herbarium, because these kind of specimen, especially their way of mode of the study, aim of the study is different, because they are going for only anatomical studies. So when they are going to collect anatomical or cytology or chemical studies, they may not collect flowering fruiting. So if it is flowering fruiting specimen only, it's helpful for us to deposit in the herbarium. That will be helpful for whoever comes to further visit that particular specimen. Easily they can identify. If without flowering and fruiting, if you collect the specimen, there is no much use for the herbarium. So these kind of herbarium especially not welcome to deposit particular specimen in the general herbarium. So these are the different kind of herbarium. So the herbarium is having the major role actually the plant conservation and taxonomy. So you can see that there are plenty of uses is there based on the herbarium. Based on the herbarium, what we can compile the floras. If you want to publish any kind of flora, for example, Andhra Pradesh flora, if you want to publish, this is already published by Professor Pullayan sir into the five volumes. If anybody want to divide the particular family Andhra Pradesh flora, you have to study the entire specimen, whatever the specimens collected and where it is deposited, those specimens you have to study in the herbarium. So maximum specimen we will get in the Madras herbarium and Hyderabad Dakhan Regional Center herbarium, Sri Krishna Devaraya University herbarium. Having the largest collection of this particular collection, especially SK University having more collection in Andhra Pradesh. So more than 45,000 specimens is having the SK University. So if you anybody want to compile the state flora, you must visit the herbarium. What are the specimens they collected? You have to document. So based on that only we can compile the floras. So like similarly we can compile the based on the herbarium specimens, the national level or international level, like this floras we can produce based on the herbarium specimen. And compilation of revisions and monographs. If anybody want to study particular family, for example, Malways, if anybody want to study that what are the specimen deposit collection, Malways deposit in a particular area you have to study before going to the field tour. Then only you will come to know that the distribution of that particular species. So these so and so hibiscus rhodesiensis available so and so in area. For example, leguminaceae, red sander. Red sander is especially endemic to our southern eastern Ghats of Andhra Pradesh, especially Chittoor, Kadapa, and Guntur. These area, Prakasam district, these area it's naturally it's available in population. So, but now after the last in 1900 also 1910, 1902 also they were introduced into the Tamil Nadu area. Now it's naturally that seed distribution is not happened. It's established some other area. When we study recently Indian studies, we find that population also. So, but anybody want to study that particular genus and family, you have to study. First, you have to consult the herbarium. What are the specimens available in that particular family? You have to write down. Then you will come to know that all the information, these particular species, where they are collected, who collected, in which date collected, in which locality they collected, which GPS readings is their particular. So based on the GPS reading, you can trace that particular species where it is available. You can go and you can recollect and you can study the further studies and like revisions and monographs. And ethnobotanical studies also we can study based on the herbarium specimen. Earlier days, when we were going to field tour, we used to write all the information in the field tour. When we are going to conduct the field tours, especially along with us, some local people also we used to take in when we are going to inside the forest. Sometimes local tribes also associate with us while going to the field tours. So then they used to ask these plants, they used to tell sometimes, sir, these plants is very useful for the snake bite. This is for scorpion bite. This is the diabetic plant. This is the very important plants for like different kinds of they used to inform. Those kind of data we used to write on the field tour. The same data we used to reproduce on the herbarium data label. These data is available at the herbarium specimen also. Whoever want to study the ethnobotanical studies, they can take these data and you can go to the particular region and consider the area. If you ask those data where they deposit the particular person name also we used to write in the field book. So when you consulted the particular evidence, you can ask the authority, sir, who collected this particular specimen, the details also is there. Who consulted the particular local tribe, the details we used to write, you can ask the authority, they will give the details also. They will share the details. Based on go and you can consult with that local tribes and you can get the data that may be really helpful for the further investigation of the things. So these are the things, major things we can produce like compilation of flora and divisions and monographs and ethnobotanical studies. Then identification of the specimen. If you don't know anything about the identification of the specimen, just you can collect the specimen and you bring to the herbarium. Just you can compare both the specimen, whether both the specimen are identical or not. So based on that, layman can easily identify by seeing those specimens. So based on that also, we can identify the specimens. So the herbarium also helpful for the identification of the plant specimen. Directly we can compare and we can identify. Then repository of the plant specimen. Because BSI or any other areas, we have plant repositories. We have n number of collections. If you want to study any kind of studies, you can take the specimen from that duplicate specimen. You can study further studies also. So climate change, based on the herbarium, we can assess the climate change. How the area climate was there, how it's responded to the particular plant those areas, those days. Now how it is responding. All the, if you study that, if we collect the same kind of specimen in different months or different years, since 100 years or 10 years, you can study the how it has changed the particular specimen with the climate change. So those kind of details also we level. I will give you one example in the next slide. So how it's climate we can study based on the various specimens also. Then plant genetic resources. We have so many wild relatives in India. It's more than 600 species of wild relatives we have in India. India is one of the center also. This wild crop relative center also. So these kind of wild relative details, all the details we have in India. If you take the rice, different kind of more than 60,000 of rice varieties we will get it. So all the earlier, whatever the earlier days rice, now almost going to be extinct. Very few people are conserving those kind of germplasm. If you go recently from Odisha, one lady, she collected more than 60 species of rice she is having in the Odisha, one tribe lady. So she collected those kind of germplasm. If you take many of the local tribes, they are collecting these kind of wild relatives of the species. If you take any kind of pulses, you will get plenty of the wild relatives in the wild. So these kind of wild relatives will helpful for us in the breeding programs for the development of the new hybrids or new cultivars. So these are the data also is available in the herbarium specimen. So if we can study the phenology study, actually when the flowering period, when this fruiting period, that is all data is available in the herbarium specimen. If you want to collect particular seeds of the particular species, you can come and visit the herbarium, then the herbarium species, when it is there, all details. When the fruit set was there, when flowering was there, all the details is there. So based on the data, this will come in the so month. So this particular month, I must go to the field and have to collect the seeds. So that's based on that herbarium specimen also, we can study the phenology study. So repository of the pollen and seed material. If anybody wants to study the paleontology studies, they can come. We used to, when we are going to collect 4 specimens. Out of the 4 specimens, 1 specimen we used to mount and deposit in the herbarium, remaining 3 as a duplicate specimen. If anybody wants to study further studies like pollen studies, anatomical studies, or paleontology studies, or any kind of studies, so we can produce, we can share one specimen. Then based on that, we can study the further studies. Then history of the science, olden days over the, because like if you take the Wallace and in Roxborough, Wallace and William Roxborough, when they are conducting field tours like Gamble in our area, especially in Madras Presidency, Gamble collected a lot of specimens from Andhra Pradesh. So they used to write on the herbarium sheet the details. So this so and so day, I have traveled from this to this area to this area. There we stayed. These are the area we have to reach. Like this, we have to 24 miles to this particular Pune, 24 miles to the Bombay. Like that they used to write. That particular details, it is available history, where they have traveled, when they have traveled, which day they have traveled. All kind of history also we used to get from the herbarium specimen. So training to the herbarium techniques. So who are going to study for the students, especially like professors, like teachers, professors like you, if you want to train the students, if you have a herbarium specimen, easily you can train the students by herbarium training, how we have to collect, how you have to make the herbarium, how you have to process, identify further, all the details you can train to the students. These are some of the importance of the herbarium. Then what are the major role of the plant taxonomy of the herbarium? So he is the person Wallace. He actually is collected so many specimens. From the more than 20,000 specimens he collected, and he made into the one catalog. That catalog we used to call as the Wallace catalog. Still we are preserving in our Central National Herbarium, Kolkata. So we have this Wallace catalog. He collected more than 20,000 species. He listed in this catalog. Just earlier there is no much description of the particular plant. This one simply used to write one name. That is the entire description of the particular plant species. This is the one line, it indicates the one species description. So he maintained 20,000 species, 20,000 specimens. He collected, he made it one catalog. These collection not only by himself, but like Roxborough collected, Gomez collected, Griffith collected, White collected. These 4 or 5 people collection they made into this Wallace catalog. At present, we have more than 12,340 specimens, Wallace specimens. We separately preserved in Central National Herbarium. If anybody want to study this particular historical specimen, you can write letter and you can come visit and you can study these specimens. So herbarium also sometimes used to give us the resolve the taxonomical problems, especially in the many cryptic species. Always no need to go to the molecular studies for identification of the specimen. Especially in the cryptic species, So these molecular studies will be helpful for the identification of the species, identification of those cryptic species. Cryptic species means this identically these species are very similar. We cannot separate it basically based on the morphological characters. But actually, basically both are distinct species. But in the morphologically, we cannot distinguish these species. So these kind of species we can identify based on the molecular studies. So if you take one pinch of specimen from the herbarium, material that is well and good for resolve those taxonomical problems. Sometimes it is not available to construct the phylogenetic trees. If we take the pinch of material from the herbarium specimen, we can reconstruct that phylogenetic tree and whatever the class we can reconnect based on this herbarium specimen. So already I told you climate change how we can study. So you can see that this Escherichia californica. This is one of the specimens of the California puppy. So this is collected from 1906 to 2009. Continuously, every year they collected this particular specimen. So there you can see the flower size, how it's drastically changing. So year to year, different. So this is the very smallest flower. Here you see there is always previous. The shape also is somewhat slightly changing. The number of petal variation also is there. Here 4 it is there. Here you can see 6 petal is there. Here you can see 6. Here also you can see 6. Here 4. Here 4. So different kind of the size and shape and the variation also is according to the climate change. The plant also expressing that character. So we have this particular specimen. How we can study the climate change also. So herbarium is very helpful for study of this climate change also. It will be helpful for the herbarium specimen. So as of now you know that this is the latest data up to 2030 I made. So now more than 1,20,000 species under the red list category. So red list category, these are species are facing high risk of extinction. In these plants are 43,557 plant species are facing high risk of extinction. In these 17,507 species, these are the threatened plants. You can see already from 120 species extinct from the wild, already extinct from the mother earth. These are the species already extinct from the mother earth. So 39 species already extinct from the wild. These are the only animals in the botanical garden in the form of extensive conservation. So then more than 3,520 species are critically endangered. These especially these three groups already these extinct, we cannot do anything for these. Especially this critical endangered and vulnerable. 3,500 critical and 6,500 endangered and 7,400 species vulnerable. These three together we can call as a threatened species. So especially we have to focus on these species for conservation program. This critical endangered species, if you are not taking any production measure, within 5 years, this species is going to be extinct. More than 3,520 species is going to be extinct within the 5 years. If you are not taking any production measure. So especially our botanical survey, the prime objective is to collect, survey and documentation of the species, and second objective is to whatever the species endemic and threatened species, whatever the species facing high risk of extinction in the wild, those species we used to collect and we used to multiply in our botanical garden in the form of extensive conservation. We introduce and we will multiply, and again we are reintroducing them to the natural habitat. So these are the species we have to focus on, this particular this critical endangered and vulnerable. In our Andhra Pradesh also more than we have 70 endemic species. In the 70 endemic species, more than 20-25 species are and comes under the threatened category. So if one professor can at least consider one species, that is a great help to our state. Not only state, country and the globe, because that is a great help to our mother nation. So these kind of species we can reproduce, we can create awareness. If anybody want those 60 endemic or threatened plants details, I will share with you. You can take that details, and wherever it is available, you can take the particular species and you multiply it and reintroduce into the national level. You try to promote our native species, not like exotic species. Because if you promote exotic species, these kind of species produce chemicals that suppress the growth of other plants, that ultimately destroy our native diversity. So herbarium also is helpful for the assessment of the endemic plants, threatened plants, rediscovery of the plant, nutriment finding studies of the any plant, invasive species assessment, and environmental impact assessment studies also. So you see, this is the Ilex gragneriana. This is already extinct from the wild. So this is actually collected by the Nilagiri Basalt Hills in 1859 by Robert White. So after repeated visit of this particular specimen, we could not find. So in the 1998, they declared as the extinct from over India from the wild. This is the specimen. So these data is available based on the herbarium specimen. That based on the herbarium details we have our earlier staff who were there, they taken this data and they went and they searched continuously 5 to 10 years that particular area. They could not find this species. Nobody has recollected this particular species and then declared as a extinct from our mother nature. So this is another species actually earlier they did it as a endangered criteria. So this is the Madhuka dicrostemon. So this is the only, now this is also collected by Robert White in 1835 from Kollam district in Kerala. So this is the only in the world, only single tree is available in the world as of now. So this is also recently 2019, Shailaja Kumari collected from the one temple, Shiva temple in Kollam district. So then she reassessed this particular plant as a critically endangered. In 2019, she reassessed it, declared. As of now, this Madhuka dicrostemon, only single plant is available in the world. So like Illegadia papilifera, this is our plant. This is especially in our southern western Ghats, this Chittoor area and like Kalrang hills of the Tamil Nadu part. These two areas, southern eastern Ghats of Andhra Pradesh and northern eastern Ghats of Tamil Nadu portion, this Illegadia papilifera is available. In Telugu we used to call it a Gali Buduga Chettu. So these also it's facing the high risk of extinction in the wild. Then we assess as a critically endangered as per the IUCN guidelines. So this herbarium specimen also is helpful for the assessment of the critical endangered species, endangered species, and we can it will also useful for the assessment of non-determining finding studies of the particular species. We studies based on the herbarium specimens, this red standard non-determining finding we study. So you can see that these are the all small dots is the population of the red standard population. Earlier it was entered into the 1998 the indigenous criteria. Recently some people they made as a near threatened. This is a very common plant available everywhere like that they made. Then again in the 2022 we researched from especially from our Nodal Resource Center and the part of this study we reassessed this particular plant as a endangered criteria as per the IUCN guidelines. So this Cycas pedomum is also endangered plant, especially it's available in the Seshachalam Biosphere Reserve of Seshachalam Biosphere Reserve of Andhra Pradesh, southern western Ghats of Andhra Pradesh, the Cycas pedomum. So we can, based on the evidence specimen, we can assess the environment impact assessment studies also. Whenever mining, who are going to propose the mining area, those particular species they used to collect, though they used to ask us to evaluate the species, any kind of risk is there that particular species, we used to give some guidelines based on that they used to propose. They used to follow and propagate that particular species. So these kind of herbarium data also will helpful for the environment impact assessment studies and invasive species studies. So these are the some of the background of the herbarium. So now how we prepare the herbarium actually. So before going to preparation of the herbarium, we need some equipment, especially digger and trowel. This is the digger and trowel. If you want to collect any kind of plant from underground, this is the tool is required for to dig that particular plant. So pruning knife for secation. This is the secation. If you normal abrasions means easily we can cut the plant, but if it's a RD or spiny, thorny or shrub means it's very difficult to cut those plants. So for this reason we need the secation to easily cut the particular plant. Our newspapers are blotting paper. These are the blotting papers. These are so to when we are after collecting the specimen to make the herbarium, blotting papers are required because to observe the water content, moisture content, whatever is in the plant, to observe those moisture content, blotting papers or newspapers are required for the before going to field. So we are going to field because going to field all these kind of things we have to keep with us and we have to go to the field. Then plant press and folders. So after making the herbarium, we have to press the particular plant. This kind of folders we have to carry it in the field. Their handles. If you want to identify when we are going to the field, we have to write each and every data on the field notebook. So for that, if you take like minute grasses and all the things, if you want to write something about the grass pikelets, you have to study with the help of the hand lens. You have to observe with the help of the hand lens. So based on that only we can write the information for micro small plant. Rope also is required. If it's a very big tree, to save our time, just you try some of the stone or something, you can throw the branch and you can take the one take up that particular plant. So knife also is very important. First aid box. When we are going to field, we don't know what kind of friends we can come across. So those kind of things for our safety purpose, we have to carry the first aid box also. So many of the time we used to stay in the forest itself. We used to go 15 days tour continuously in the one tour is the 15 to 20 days we will be in the forest. Sometimes we used to come to the nearby habitations. There we used to stay. Otherwise, sometimes continuous stretch 5 days continuous stretch we used to go. There that time we used to sleep in the forest only. For that reason, tent is required to stay in the forest. So among the all, if we are not there, we can manage. But this is the very important thing is the field notebook. If it is not there, it's a very difficult because all the data, whatever things is data is there, we have to write in this particular field notebook. Simply we are going to not going for enjoyment purpose. We have to collect all kinds actually that particular plant belongs to which district and belongs to which status and which state you have collected. What is the particular plant name? If you know the scientific name, you can write. If you don't know at least family level, if you identify, you can write family level. If you don't know scientific name and family name also, you can write local name and locality, where you have collected, in which district, which mandal, which forest, kind of forest, which reserve forest you have collected, that details you have write. And GPS regarding readings of the particular plants you have write. Habit of the particular plant. What whether it is hail, bird, shrub, a climber, a tree, or lion, or that kind of habit you have write. Height of the particular plant. What is the height of the particular plant? Vegetation. What kind of vegetation is there? Where there is a moist vegetation, dry vegetation, scrub forest, or paddy forest. Those kind of details we have to write in that field notebook. So what kind of associate plants it's having that particular species? Because some of the species will grow only in the particular association of the particular species. So those kind of species details are you have to write. And distribution. Whether it is a common species, it's a frequently it's available, it's a very rarely you are finding, or very occasionally you are finding, these kind of details you have to write. Abundance of that particular plant also you have to write. And significant nodes. So this is the very important. When you are saying the when you are seeing the specimen, what kind of characters, strikeable character, whatever is there, that you have to write in the field notebook. Then if you know the any kind of uses, like already I told you, we used to take like local tribes, they should give the information of the particular uses plant. So those kind of uses also you can write. Then collected by whom? Who collected this particular plant and identified by whom? So these are all details we have to collect when we are going to the field. So the same, but this is the one field notebook is having a 100 numbers, that is starting from 1 to 100 numbers. The same kind of number 6 to 7 detachable tabs is available here. So these tabs we have to tie with the thread. The same one tab we can pair it and we can tie with the headband, whatever we are collecting the headband specimen, with that we have to tag it. So that is easily we can verify that particular same kind of number. You can see the 5901, here also 5901, 5901, same kind of number. So the particular specimen data, 5901 data, here it's available. This based on this, we can write when we are writing after identification of the specimen, when you are making herbarium, there we have to write these kind of all data. So then GPS, nowadays GPS is very important. To map the particular species, GPS reading is very important, and camera, take the photographs, camera important. So when we are collecting all kind of plants, if you are not carrying this material, especially these chemicals, then whatever you have done work is waste only. Because and the next day the all skeleton will come out, all leaf, all flower or leaf pods will be discharged from the particular specimen. So for this you have to preserve that particular plant specimen. So for preservation, alkyl and formaldehyde and acetic acid and 7:2:1 ratio you have to prepare it and you mix it and you take the element while going to the field. You can carry in the bottle and just you pour it, you preserve that particular specimen 30 seconds. Dip it will be sufficient for the preservation of the specimen, especially to stop the formation of the obstruction layer. So these are the things we have to carry. So if you want to retention of that particular flower, like a blue color, blue, reddish color, red, as if you want to maintain, these are the proportions you have to use. If you use these proportions, then the color will be retained as it is only. So this is the material is required before going to the field tours. So when we are going to the field tour, the first step is the collection of the plants. So before going to the collection of the plants, actually you have to decide actually what you want to study. Whether you are going to collect general flora or any biodiversity assessment, or any gap areas, or any special group you are studying, or any invasive species you are studying, or endemic species you are studying, that you have to decide your aim. And decide about your area, whether you are going to unexplored area or explored area, or protected area, any kind of area you are going. If you go explored area, you will not get anything. If you go unexplored area, you will get plenty of new things. So this particular, before going to the select the topic, you have to choose the area, which area you want to study. Then before going to the field, you have to secure the funding also, because field tour is not easy. If you go conduct one field tour, it will take, if you go 20 days, at least it will take 30 to 40,000 rupees. So before going to field tour, these all kind of things you have to see. So the main objective of the collection of the plant is to build up the new herbarium. Or if anybody don't have herbarium in their college or university, for the development of the new herbarium, they can go to the field to collect the plant specimen. Other like we are having all different kind of herbarium. Even though we are going to field, why we are going to enrich the collection of our existing herbarium collection. So if you want to write any kind of flora or division or monographs, you must go to the field to collection of the plant specimen. So introduce any kind of endemic plants or threatened plants or critical endangered, whatever is the plants or economically important plants or wild edible plants, you can bring those plants and you can introduce to the garden. For that also you have to go to the field. to carry out any kind of research, like already told, analytical studies or psychological studies or biological studies or embryological studies or phytochemical studies. So whatever the studies if you want to do, you must go to the field to collect the specimen. After the collection only you can study whatever you want to study, that you can carry out. So for this purpose, you can go to the field. So ethnobotanical studies also we can go to the field. If you want to give training to your students also, you need specimen. Without specimen, you cannot give to the training to your student. For that also, we have to go to the field for the collection of the specimen. So when we are going to the field, actually what we have to collect. So what not to collect, what we have to collect, that we have to decide actually. Always the collection should be made from the healthy plant. The tree of the it should contain either flower or fruit. If it's a flower and fruit is not there, means there is no use. In case of grass or sedges, the old plant we have to collect because the underground portion is very important, especially in the grasses and sedges. So the underground portion is whether it's annual or perennial, it will come to the base than that underground portion. So that is a very important character for identification of the, especially in the grasses and sedges. So collection of the variation between the species. If you collect some kind of plants, if you show the yellow color and same plant if you show the white color, those kind of variation you have to collect and you have to record in the your field note. If whatever the specimen you are seeing, don't color, at least 2 to 3 number of specimens is more than sufficient. So the standard size for the specimen is a.
Excuse me, sir. Sir, it's already 4:40. And our session should end by 4:45, sir. We have validatory followed by.
So, madam, when I have to finish, how much time I have? Shall I conclude?
Time given to you is completed, sir. 1 hour we have given.
How much time, ma'am?
Now it is 4:42, sir. Another 3 minutes you have, sir.
15 minutes.
No, sir. Time is up, sir. Already 1 hour we have given, sir. So, sir.
Stop, ma'am.
1 hour, sir. From 3:40 you are talking, sir.
Okay. Should I stop, ma'am, now?
Yeah, we have validity, sir. Our commissioner will be joining us by 4:45.
Okay, then. Okay, I'll stop, ma'am.
Please conclude this, sir.
Ma'am?
Conclusion, sir.
Conclusion, ma'am, because actually here you starting, yesterday he told me 2 hours. Then actually topic itself not knowing. Actually whom have to teach, I don't know. Then today morning, in afternoon I came to know 1 hour. Now it's
No,
like
actually for every resource person we have given only 1 hour, sir. Maybe.
Okay. So this actually okay. After collection, actually we have to go for preparation of the herbarium. So preservation is very important. Already I told you why we have to preserve. If you are not preserving a particular specimens, all parts will be detached. So there are 2 kind of methods: dip methods and wet method. So dip method means we have to the specimen, you have to dip it in the 30 seconds in the whatever the solution is prepared in that you have to keep dip into the 30 seconds. Then you can keep it in the blotting papers. If you are going long-term project, 15 to 20 days source is very difficult to change every day the plant specimen. For that reason, you have to follow wet method. We can make it into the bundle and you can pour whatever the mixture 7:1 ratio, pour it in the P side of that particular bundle. So then you can keep those bundle into the polythene cover so that the specimen may not get any kind of damage. Whenever you get ready after 6 months also, you can take those kind of specimen for the further side. So once the specimens preserved, then you can go for the pressing the specimen. For pressing, main important is the plot in that particular specimens. So then when we are going to the pressing specimen, we need to take some special kind of characters. Here when you are collecting these bulky seeds, so we have to make separate. Then we are after once this pressing is over, then we can go for the drying method. So sun drying is a common method for the drying. Then after the once drying is over, we can go for identification. Once is identified, then we have to go for poisoning. Once poisoning is over, then we have to go for mounting. Then after mounting, we have to go for stitching. Then when we are going for stitching, then we have to go for labeling. So once the labeling is over, then we have to go for index and ledgering and accession. Then we have to interpret the specimen. This species cover is specimen is ready. Once the specimen is ready, then we have to place in the species cover, then genus cover. Then after that, we have to go for interpretation the particular family level. So after this interpretation is over, then a lot of the maintenance part is there. So whatever the because more silver fishes and all, it will come and attack. Those kind of for maintenance, we used to give methyl bromide and phosphine and carbon disulfide and carbon tetrachloride. We used to give for the saucer we used to keep for maintaining of those herbarium. So these are some of the general instructions. So this is the methodology for the preparation of the herbarium actually. So the virtual herbarium it will take at least 20 minutes. I may not now. These are the procedure actually. this one chart I will explain. When you are taking the specimen, when specimen is ready, then we have to, before entering the digital library, we have to see the all the whether nomenclature and everything data is there to correct or not. We have to check. Once the data is ready, then we have to capture the image. Once the capture, then you have to enter the all data. Then once data entered, then we have to make the three files, and the zip files, JPEG files, and small IDH files. These kind of IDH files we have to upload into the website, and then we can export to the digital library. So these are the methodology how we have to do. So we have Indian Virtual Librarian, we Botanical Survey of India separately. We have a Virtual Librarian. This is the website idh.bsi.gov.in. Whatever the all collections, it is there in Indian Virtual Librarian or Botanical Survey of India. Just you can go and you open it. All kind of specimens available. So like this details it will come. If you enter this all the scientific name, who are the collector, all the plant details it will come. So like this, all details it will be there. So this is the data we process. Like this data, whatever if you want to collect data, you will get. If some of the online websites also is there, like Plants of the World Online, if you get the name, you will get all the plant names, like Plants of the World Online also is there. Just if you enter the name, all kind of specimens, whatever is available in the world, you will get. So few catalogs are available. Then World Flora Online also is available. There also you will get. For our Indian perspective, so many virtual experience also are available. So this actually it will take more than 20 minutes to explain it to the virtual experience.
Thank you very much, sir. Sir, we have received your mail ID and phone number, sir. The participants have any doubts? I think one or two may ask, sir. Otherwise, I will share this mail ID.
They can ask.
Please.
Good evening, sir.
Good evening, sir.
We have a small doubt that one. So, is there any possibility to upload the pictures of our plants nearby our areas on our website so that it can be easy to study from the remote areas for the research purpose?
Sir, you are not getting. If, do you want to upload your picture to our website, or do you want to images of our data to you?
From our side, for example, we are living in this TOT states. The session will be one of the.
Actually, you are not properly audible, sir.
Sir?
Do you want specimens or images of ours? Do you want to?
Pictorial distillation. So when we take the pictures of nearby plants, if you upload in that website, you can have it upload of India website like that. It is service website, so that it can be easy to collect the pictures and to study the difference between the species of various areas.
Sir, what you are saying, you are not at all properly audible to me. My mail ID is swami.2706.
Yes, sir.
You can that way, whatever you want questions, I can give you answer. Because you are not audible properly. What kind of you are? Whether you want specimens of us, or you want to images from us, or you want to upload your images in our website.
Yes, I will contact you.
My mail ID is swami.2706@gmail.com. If you have any question, you can type. You can send me, sir. I'll give answer to you.
Yes. Yes. Then there is no other queries about process. I will let me do this session by to our professor. Yes, what is that? Sideways in Botanical Survey of India. Welcome. We will answer this. Going to be starting the current study because two examples are very rare nowadays. So the entire session gone through excellent slides and good pictures. And we are also happy to see some of the plants in India here, such as middle insiders, like that. These plants are very not at the same time, we also know so many applications. Sometimes that's all we can and consider development system. We have a proper of students, we complements as well as knowledge sharing, like elements from the classes and projects. Our appreciate office, we also try to develop for the stakeholders. Seminars as well as resource person driving programs for the sessions. So, speech is also excellent, sir. We have to do this which are related tracks. The same as methodology, very different to we'll come back to thank you very much for providing. Thank you. Thank you very much. So thank you, sir, for the message. Thank you, sir. I have purchased.
Swami sir, there
Ma'am.
was some technical error, so I think you were not able to hear the voice of the participant.
Your voice is clear, ma'am. Your voice is clear.
Thank you. There was some issue at that end. Actually, Dr. Bakshu has proposed a vote of thanks on
Okay,
behalf
I
of
thought
all
something is
NRCs.
asking, unable to hear what is asking.
Yeah. So on behalf of all NRCs, we really thank you for that excellent presentation, sir. And as you extended your help from BSI, that if we write a mail for any extension activities for the herbarium, I think most of our college faculty will definitely seek help for that, sir.
Okay. You can
Thank you
write
for
anytime, ma'am. There is no issue. You can consult our Dr. Regional Center Herbarium. That is a very, because now whatever the collection from Andhra Pradesh, especially it's available in Hyderabad. You can consult Dr. Rasingham sir. He is the head of office in the Deccan Regional Center. He is a very cordial person. You can go and consult him. All kind of herbariums. Almost we digitized all herbarium specimen in Deccan Regional Center.
Okay, sir. So one more request from the participants is, can you please share your PPT? Because
I'll make
the
in
preparation
PDF. I'll
of
share mine
herbarium and
because this
all.
is more than 112 slides is there. I'll make in one
Okay, sir.
PDF and I'll share.
Yes, sure. Thank you so much, sir.
Okay.
It's a great honor to have you on this session. Thank you, sir.
మేడం చేసేయండి చెప్పండి అది మేనేజ్కి వాళ్ళే చేస్తారు కాబట్టి మీరు చెప్పేయండి మాకు వస్తుంది కాబట్టి అందరూ NRC లెవెల్ వాళ్ళు డైరెక్ట్ చేసుకుంటే సిసి నుంచి ఎవరు రారు మేడం ఫోన్ చేశారు నేను ఈ బ్యాటరీ back response.
Hello.
Successfully complete చేసినందుకు
మేడం NRC తణుకు ఇన్ఛార్జ్ ప్రిన్సిపల్ సార్ మాట్లాడతారు మేడం.
గుడ్ ఈవినింగ్. NRC తణుకు నుంచి మాట్లాడుతున్నాం సార్. ఈ సక్సెస్ఫుల్ గా కంప్లీట్ చేసినటువంటి ఈ 5 days TOT ట్రైనింగ్ అందరికీ కూడా థ్యాంక్ యూ వెరీ మచ్. మన కమిషనర్ గారికి, అలాగే మా కళాశాల ప్రిన్సిపల్ గారు అనిల్ కుమార్ గారికి కూడా థ్యాంక్ యూ. NRC పరిధిలో వ్యాలిడేట్ చేసుకోమని తీసుకోవాల్సిందిగా రిక్వెస్ట్ చేస్తున్నాం సార్. థ్యాంక్ యూ సార్.
We thank all the NRC coordinators, principal and academies for organizing this 5-day TOT in a successful manner. As per the instructions received from CCE, they have requested us to conduct the validity with the NRC principals. Thank you very much. Thank you one and all. So we are going to end the online session.
Okay, ma'am. Thank you.